This is lecture 2, I think, 2 or 3 on genetics. And no, today's 3. Today's lecture 3, genetics. And we're just going to pick up where we left off. We left off on this slide last time. We're talking about ways in which we can screen ourselves for genetic anomalies. And there's 2 basic kinds of invasive testing, right? So there's non-invasive testing. You can do blood tests, you can do ultrasounds, even though technically ultrasound is not invasive in that you're breaking a body, cavity, or you're not penetrating the skin. You're sending disruptive waves of high pitched frequencies into the tissue. That's got to do something, right? So briefly, amniocentesis is when we have a developing fetus about 14 to 16 weeks old, and it's surrounded by this very thin membrane that has a fluid in it called the amniotic fluid. So this is the amniotic sac. And what we do is we take a needle and we using ultrasound to guide it, we very carefully bring the needle into the womb and we withdraw a bunch of this fluid. The fluid contains both, then we centrifuge it lightly. The fluid contains some cells, some fetal cells that we can culture. These are living cells that we can culture, and they take a long time. They take a few weeks to grow up enough so that we can do a karyotype. In the meantime, we can take the fluid and do some biochemical tests. And then we can also do additional testing on these Petri dish ones. So overall, amniocentesis is more comprehensive. It's more diagnostic. It just takes a long time to do. All right. So you do an amniocentesis if it's not too concerning, but concerning enough. The rate at which I should say the risk here is that there's a very, very small increase in the chances of spontaneous abortion or miscarriage. But it's less than 1%. If there is an urgency that needs to be taken into account, we can do what's called CVS. We can do this much earlier in pregnancy, between 8 and 10 weeks. And in CVS, what we do is we take we aspirate a very small amount of tissue directly from the placenta by going up the vaginal canal into the cervix and with a catheter. And we can take these little bit of cells. It's almost like a biopsy. And the benefit here is we can do an immediate karyotype. Just a few hours later, you can know the karyotype. And you can do some biochemical tests. The drawback here is there's less you can do. There's fewer tests you can run with this because we don't have access to the amniotic fluid. But the benefit is it's immediate. And then the other problem is that it causes a slightly greater increase in spontaneous abortion. It's more than 1%, less than 2%. So it's still small, but if you spend a decade trying to get pregnant, every little 1% helps. So that's that. That's all I'll say about that. Wow, that was annoying. Okay, there. So now the key here is when you do a karyotype, remember, you can't really get beyond the gross chromosomal abnormalities. So you can see chromosome counts and you can see huge swaths of chromosomes missing or not. But that's it. You can't see individual mutations, right? Just keep that in mind. And then going along with the end of the last chapter, we talked about the diversity between different organisms. That's bio 2. But just as kind of a preview here, our XY system is just one of several kinds of systems in the animal kingdom. There's the ZW system in birds, in which the hemizygous state is swapped with the genders here, with the sexes. So the male is the one that has the homozygous, the diploid state of the sex chromosomes. So the female is the one that carries one of each. And then in insects, there's a few. There's a couple. There's the exosystem in some insects, like you see here in crickets. So the exosystem is you either have one X chromosome or you have two X chromosomes. So there's no Y. It's just X. And if you have one, you're male. If you have two, you're female. So it's kind of like our system minus the Y chromosome. So think of it, there's the XY system and the X nothing system, just the XO system. And then there's the haplodiplo or haplodiploid system. And this is in honeybees. And you can see here is if you're haploid, you're male. If you're diploid, you're female. And so like the queen bee would be diploid and the males would all be haploid. Okay, so that's that. So what we're going to do now, we're going to enter into this idea of going beyond, but technically this title slide should actually be moved. Chromosomal inheritance, we're going to talk about with Morgan. But before we do that, we're going to learn about just a little bit of what are called multifactorial genetics and some of the violations of Mendel within unifactorial genetics. So what I mean by that is in genetics itself, you can divide genetics itself basically into two branches. Unifactorial genetics concerns itself with what we've already learned. One trait associated with one gene, that one gene has a pair of alleles with one being dominant and the other being recessive. Right? Very straightforward. And really that's the exception to the rule. That's as simple as it gets, but it's not common. Okay, so even single genes can behave in this non-Mendelian manner, right? And so I'll say that again. When we say a Mendelian trait, we're talking about a unifactorial trait. Okay, those are synonymous terms. We're going to do codominance, incomplete dominance, multiple allelic traits, and pleiotropy. Right, briefly, right? Codominance, you can think of that as a both win. When I say both, we're talking about that pair of alleles. Okay, neither is dominant to the other. They are both dominant, meaning they are both expressed as the phenotype. All right, so you can think of Mendel's green and yellow peas. Imagine a marbly swirl of green and yellow in which they were even. That would be codominant. Okay. I'll give you some examples of this soon, but I just want to hit these into your head. So codominant, both phenotypes are expressed. Okay. Incomplete dominance, neither wins. It's a compromise. It's incomplete. Okay, so this is a compromise. This would be like yellow and blue peas making green peas, right? This would be something in the middle. And I'll give you examples of this one too. So incomplete dominance means there is no dominant allele. Codominance means they are both dominant. And so what's dominant mean? Dominant means you see the phenotype. Okay. If an allele is a dominant allele, that means you can see it. If it's recessive, you can't. So if they're both dominant and there's no recessive allele, that's codominant. It just makes sense now when you explain it that way. Okay. A multiple allelic trait is a trait where there's one gene, but there's more than one allele. So there's not just dominant recessive, there's like 1, 2, and 3. And then lastly, pleiotropy is when one gene literally controls another gene. Like the point of this gene isn't to make an enzyme or to make a pigment or whatever. It's make an enzyme to make a pigment. It's to make a gene, to make a protein product that literally controls this one somehow. Okay, so we'll go through what could the pleiotropy is. I'll give you an example of that. Right, and then I'm sorry. Oh my gosh, I'm so tired. What I just explained to you is epistasis. Epistasis, epistatic gene control. That's one gene controlling another gene. I'm sorry, you guys probably just wrote all that down in your notebook. So cross out pleiotropy and replace that with epistasis. The definition was right. Epistasis. And that's down here with multifactorial genetics. So pleiotropy is when one gene controls many traits. Okay, so that's a simpler concept. So one gene, many traits, trait 1, trait 2, trait 3. Okay, you see it has a pleiotropic effect. All right, so that's a unifactorial, Mendelian, non-Mendelian. All right, multifactorial genetics where there's more than one gene involved, basically. So epistasis is when gene A controls gene B, generally in a negative way. Generally, you would say gene A turns gene B off, right? And so if gene A has different alleles, some of those alleles work, some of them don't. You can see how there might be a variation there. Okay. Polygenic inheritance means there's multiple genes controlling one trait. So you have all these genes controlling one trait, right? So the example of that would be like skin color. or body height. And then lastly, sometimes you've heard the old adage, nature versus nurture. All right, so sometimes the environment plays a role in our outward phenotype. So that's the basics here. We're going to go through some examples now. All right, so here's codominance. There's a flower called a camellia, and you have white camellia and red camellias. And if you cross a white and a red camellia together, okay, so pure breeding white, pure breeding red, you don't get white or red, you get a little of both. Okay, so you see how the parts that are white are just as white as mom, and the parts that are red are just as red as dad, or vice versa. Okay, codominant. There's two phenotypes. Don't look at this as a phenotype. Okay, that's two phenotypes in one. And the reason I say it like that is because when you do, let me just skip ahead really quick. When you look at incomplete dominance, okay, so roses, you cross a red rose and a white rose, you get a pink rose. Okay, this pink, that's a third phenotype, right? And it's a phenotype that's somewhere in the middle here. This is what we used to see as blending. It looks like blending, right? The difference is, and the reason it's not actually blending, is if you take this pink rose and allow it to self-pollinate or cross-pollinate two pink roses together, you'll get this 2-to-1 or this 1 to 2 to 1 ratio. You'd get red to pink to white of 1 to 2 to 1. In other words, each of the genotypes has its own phenotype. All right, so remember, the Mendelian pattern is 3 to 1, right? The monohybrid cross gives us a 3 to 1 phenotype. So this is a violation of the 3 to 1 phenotype. This is a 1 to 2 to 1 phenotype. Okay, so that's incomplete dominance versus codominance. Now let's go back to codominance for a second, because there's a very important example here. Our blood typing is a good example of both codominant and multiple allelic traits. All right, so here's how blood typing works. It's called the ABO system. All right, there's other blood systems, and there are other important antigenic markers in our blood, but this is the predominant one. The other one being the Rh factor, and you're either plus or minus with that, but we'll talk about that briefly at the end of this. All right, so here's how it works. You got two alleles, right? You have two alleles, you got one from mom, and you got one from dad. All right, so here's the gene, and either that allele can either be an A, or it can be a B, or it could be an O. All right. One of the ways we do the nomenclature for this is we do a capital I versus a lowercase I, and if it's capital I, it's an A or it's a B. All right, the way you do it this way, this shows you that the lowercase I is the recessive trait, right? Capital I is dominant, lowercase I. So this generally associated with O, this would be B, and this would be A. So using this system, this is more informative. This tells us that A and B are functional products of some kind, right? It turns out if this is a red blood cell, there's a surface marker, a surface antigen. It's a glycoprotein, and we call it A, or it's B. All right. If the gene itself is mutated and it doesn't produce a viable protein product, we call that O. All right. So a blood cell from a person that's O-type blood would look like this. A person with A-type blood would look like this. A person with B-type blood would look like this, and a person with AB-type blood would have a bunch of them, some of each. That's why it's codominant. Both the A and the B are expressed. Okay, so now that you understand that, you can mix and match these three alleles, right? You only get two. You get 2 out of 3. Draw 2 cards, and here's 3 cards, right? So think about it. You can either be, and so let me just to doodle it, I'll make it simpler here. You can have 2 A's, or you can have an A and an O, right? You can have an A and a B, or a B and an A. You can have a B and a B. You can have BO, which is the smelliest one, and you can have OO. And I snuck that joke in there. Right? So if you look at these two, and you look at these two, both of these will give rise to either the B or the A phenotype, right? You just need one. And then these two will give rise to the AB phenotype. And then only the double O will give rise to the O phenotype. Okay, So that's how blood works. As for the plus and the minus, there's a separate gene controlled by us. It's a basic Mendelian trait. It's called RH. It stands for it was discovered originally in rhesus monkeys. It's called the RH factor. It's a ammonium transporter in the red blood cell. So it sits here and it allows the pumping, really, it allows the pumping of ammonia. And you either got it or you don't. So think of it as a classic Mendelian dominant recessive. So, you're either Rh positive or you're Rh negative, right? And so you can imagine do a basic Punnett square with plus and minus, basically. So if, a woman who is homozygous dominant marries a man who is homozygous recessive, right? All of her children are going to be dominant, right? So the problem arises when you have a mother who is Rh negative, right? So here, actually, let me give you the problem scenario. You have a mother who's Rh negative, and she marries a man who's Rh positive. Let's say he's dominant for it. All right, so female up here, male along the side. So there's 100% chance that the kids are going to be Rh positive. Her immune system has never seen Rh before because she's Rh negative. All right, so the way immunity works is when you're developing in utero and you come up as a baby and you start growing for the first year or two of life, you're basically building out your immune system, your T cells, your B cells. Your B cells produce these things called antibodies. Antibodies are these small little proteins made. Oops. Let me use whiteboard. Really, one wrong click and you lose everything. Whereas whiteboard, that does not happen. So let me just share my screen here. Whiteboard. All right, there we go. I'm doing viruses in my other class. Let's go over here. All right. So a woman is Rh negative like this. And she marries a guy that's Rh positive, right? And then so all the kids you can see here would be positive. That's the phenotype positive. Right? Okay. So what happens is antibodies. An antibody is a protein produced by B cells. It consists of a heavy chain and a light chain, one of each. Basically, you can cartoon an antibody like a Y. The key is this region right here is called the variable region. It is a random shuffling of genes. So it's kind of like crossing over, but it's happening only in B cells and only in this one genetic region. It's really cool. And so every single B cell in your body, billions and billions of B cells, produces a completely unique antibody that's completely random. This is how we're able to defend against the unknown. Okay, and so a woman who is born Rh negative has let me add one more thing to that. So when you're born and during your development, think about the idea, since it's all random, that some of those B cells are going to produce antibodies that are specific to proteins in your body. That's bad, right? And so what happens is during the development of your immune system, your body is killing the B cells that recognize self. Okay, this is the most important point in immunology. We are born with no B cells that recognize self. If we are, we get autoimmune diseases. That's part of what autoimmune diseases are, is that we produce antibodies against ourselves, and that's bad, right? That's why you have inflammatory responses. So, I normally get sick. But hey, let's talk about COVID. All right. So let's say you get exposed to this virus for the first time. No human has ever been exposed to this virus. It's new to the world. It's a novel, it's an emerging infectious disease. And so nobody is immune to this thing. Nobody has a competent immune response to it. So what happens is you get infected with it. It starts producing viral particles. It starts shedding its proteins everywhere. Your cells are dying from it. And so there's some B cell somewhere in your body gets handed this random fragment of the S protein from the virus. And the B cell's like, hey, I know this. It's a random B cell that happens to know this thing. And so that B cell goes through some stuff and it starts dividing and it starts producing massive amounts of itself, right? Lots and lots of mitosis. So you have thousands and thousands of new B cells that are all that B cell. That takes about 2 weeks. This is called your primary immune response. And after that 2 weeks is over, your blood is filled with these things that are producing antibodies everywhere, thousands per second, thousands of antibody molecules per second at its peak. And that's the idea of immunity, right? That's the idea of the immune attack. It's called your adaptive immune system.
And like, is that kind of why, like, after you're infected with like coronavirus, you can't get it again for a while?
Well, yeah, right. That's
Because the
true with any
antibodies
infectious.
is kind of fighting off, even though it's not present. So if it were to be present again, it would just fight it.
Okay, so if you think about just again, this I wish I could teach you guys immunology, but just briefly, here's the amount of antibody in your blood. You got nothing, and then 2 weeks later you got a bunch, and then slowly it goes down like this. Okay, so a few weeks later after the infection, you don't have any more antibodies in your blood. What you do have are what are called memory B cells. So some of those B cells through mitosis randomly become memory cells. They're not out in the world doing antibody production. They're waiting in your lymph nodes, and they're going to sit there and remember. So the next time you get infected, right, then the virus starts going through its cycle again. Your memory cells are like, oh, I got this, and they very quickly respond, and that's called your secondary immune response. And so what happens there is you get a very, very fast, rapid increase, very fast, massive increase in antibodies. And they last in your blood for months. That's your secondary immune response. That's immunity. Okay, the problem, there's all sorts of ways that things can evade this though. For example, the virus can mutate, which happens a lot, very fast.
Right, like how there's different strands. How about that?
Right,
Does that
strains.
not work
Right.
for
Yeah,
different strands because it's
so
slightly different?
that means that initial antibody is not going to recognize the new strain necessarily. Right? Particularly if the new strains have a problem such that their S protein, their spike protein mutated, so they're unrecognizable. And actually, handily enough, actually, if I go over here from my microbiology class lecture. So the spike protein basically looks like here. If you go over here, see the little sun thing. The spike protein has a little stick sticking out. This is an enveloped protein, an enveloped virus. And so if the genes that encode for these spike proteins, if this suddenly changes into a different shape, now the antibodies that you spent all that time coming up with and making are useless, right? So you have to go through a whole thing again. So that's how you can get reinfected by a different strain. And this is how the flu works. Influenza is horribly mutable. It mutates regularly. So every season, every flu season, we have to get a different shot because it's always changing. All right, so that's the basic stats. Let me go back to the PowerPoint now. All right, and so the idea is the first, when a woman is Rh negative, okay, what that means is her immune system has never seen the Rh protein, which means she's got antibodies against it, right? She'll fight it. So her fetus in her body, which is Rh positive, is producing this weird protein that it's never seen before. Now, here's the thing. The first pregnancy generally is fine. She's developing her immunity to Rh. The second kid is where the problems start, because now her body mounts a much more powerful secondary response. And that's where the kid's blood cells are getting, it's called hemolysis, where the blood cells start getting lysed. The good news is we have treatment for it now. Back in the 70s, we learned that if we inject a woman who has this going on, we can inject her with antibodies to Rh. And I'm sorry, not antibodies to Rh. We can inject her with the antigen itself, and we can basically, it's kind of like getting allergy shots. It tempers the immune response to the Rh. So now we can have children being born where the mom and the kid are different. And you can totally blame dad for this. Because he's the one that came with his Rh positive genes to the mess. All right, so that's an example of codominance, and that's an example of multi-allelic trait. Right, multi-allelic. There's the A allele, there's the B allele, there's the O allele. Right, so there's this allele, there's this allele, and there's this allele, 3 alleles, more than 2. Okay. All right. This is called the agglutination test. The idea here is, imagine you had a microscope slide and you put your red blood cells on, and you want to know what your blood type is. Okay, so what are your possibilities? Again, you have A or a B or AB or O. All right, now imagine I sprinkle some, let's say that this is A-type blood. Let's say we know, you and me know it's A-type blood, but the test doesn't, right? So it's A-type blood. Imagine I added antibodies specific to A, right? What that means is the antibodies are going to stick to the surface of these cells. And when the antibodies, because they're Y-shaped, right? They're going to be able to cling to multiple cells. And then actually, some antibodies form these kind of really cool structures like this. Think of it like a dust bowl, where they can form these aggregates. So each one of those can bind to a red blood cell. Okay, so they'll clump together. You see that? So this is called agglutination. Our blood does. It's not clotting. This is agglutination, when proteins cause these clumps to happen. So it's called an agglutination assay. And if you get the clumps, that means you get a positive response. That means that's your blood type. So go over here, you can see this. A-type blood. Where's my mouse? A-type blood. All right, so you can either get either of these genotypes, and if you take, so the body will make anti-B antibodies, right? Think about it. If you're A-type blood, you know A, you're going to fight B because you don't know B. Your immune system doesn't know B, so that's an invader. So the antibodies made by the body will be anti-B. All right, and then separately, if you did the agglutination assay with the anti-A antibody, you would see the clotting, the agglutination. You'd see the clumping of the cells. If you treat them with an anti-B antibody, you won't see anything. Okay, and actually, in my immunology class in college, I remember now, we actually did this assay. You take a little finger pinprick, a little tiny prick, you get some blood off, you put it on a slide, and then you have, little tubes that have the A and the B on it, and you add the little antibody to it, and then you look it under the microscope. You can see it exactly, so you can blood type very easily. All right, so just look at this. All right, AB blood will clump with both. B-type blood will only clump with this. O-type blood won't clump at all. Okay, so that's that. So now you can see why we have universal donors and receivers. So if I have B-type blood, so I have B-positive blood, right? So if I'm in the hospital and I need a blood transfusion and they accidentally put A-type blood in me, my body's going to mount an immune response to it and blow it up. So that's not going to help me in any way, right? If I have AB-type blood, I can get any blood. That would be the universal acceptor, right? And if I have O-type blood, I can't get anybody's blood except other O-type blood. Okay, whereas I can give my blood. Go ahead, Taylor.
We want them to get like the chunky pieces.
We don't. Oh, okay. So if this happens, right? So if I have B-type blood. If you put A-type blood into my body, right? If I have B-type blood, that means I produce antibodies specific to A. So I'm gonna kill it, right?
Okay.
So that's why.
Okay. That's what I thought. I don't know. Never mind.
Well, so here's the thing. When you learn this for the very first time, the reason this is confusing is, or rather, it's not confusing if you just keep this very firmly in mind that whatever you are, you will fight what's not you. If I am B, I will fight A. If I am AB, I like everybody. If I'm O, I hate everybody. Okay, that's the key to understanding immunology in the basic levels. Right? And if you don't really have that in your head clearly, then you get really confused. I have B-type blood, so that means I have B-type antibodies. No, it means you don't have B-type antibodies, right? You shouldn't have B-type antibodies. If you did, you'd be dead because your blood wouldn't be there, right?
Because it would get all big, right? Like how these ones are all like clumpy?
Because it would clump up, yeah, and then eventually the cells would get lysed, right? This would happen. Agglutination would happen.
Okay.
You got it. All right, now add to this, you can be A positive or A negative and B positive and B negative and AB positive and AB negative and O positive and O negative, right? So now the true universal donor is O negative blood, and the true acceptor, the universal acceptor, is AB positive, right? AB positive blood can accept any of the other kind of blood types. O negative can only accept O negative, right? And so people who are O negative, those are the ones that the blood banks want. regularly because that's the kind of blood that everybody can get, right? I had a student one semester many years ago. He was probably like 6'4", 6'5", and like really big guy, huge guy. And when I taught all this in class, he's like, yeah, they love me there. I'm O negative. He said one time they took so much blood out of him, he started getting dizzy because they could take so much blood from him because he was just such a large person, right? He wasn't fat. He was just like Hulk. Like he was just huge guy. And they would take a lot of blood. They loved him. He said they'd give him cookies and lollipops and
O is like recessive, right?
say again.
Like O's like recessive, isn't
No,
it?
so there's nothing. Here's the thing. That this is the classic example of codominance. There is no
Okay.
recessive, right? If you have the allele for something, it will express. The key is the O-type blood. The reason there's nothing to express is it turns out the gene is mutated. And so it doesn't express anything. There's no functional protein product.
So like my mom is O type. I don't know what my dad is, but my sister has O blood, and I don't know
Okay.
what I have.
All right.
So
So
I
if your
thought,
mom, ready? So if your mom is O-type blood, that means that she has to be this. She's homozygous recessive. So
Yeah.
she's like the test cross. If your sister is O-type blood, that means your dad has to be at least that.
right.
And so what you are is you are definitely like, this is you right now. You don't know what the X is. If X is also I, then you have O-type blood. If X is A, then you have A-type blood. If you have B,
Yeah, they don't bother me there. They like, my mom always says like that they're like vampires. Like they're always calling her like.
well, that's separate. That's if she's she could have Raynaud syndrome and be thin. Like thin people tend to be very cold, because they shit heat very fast. And the female body tends to be cooler than the male body, again, because it has a smaller density. The bones are a little thinner, and that's why women always tend to be a little colder than men. And that's actually why in the workplace, this is actually like a gender equality issue. The gold standards of OSHA, the workplace rules, are based on the male body wearing a suit. These like all the temperatures were established back in like the 1950s. Men in suits. So like being 68 degrees, that's terrible for most women, right? You don't want it to be 68 degrees because now you're wearing your 18 different layers to work and you're shedding and go. Whereas the guys are all fine. That's because we're in our suits and we're bigger on average and we are warmer. That's also why when you look at wedding photography, weddings in the winter, the guys are comfortable, and weddings in the summer, the girls are comfortable. So
is that also why like young high school guys always wear shorts in the winter and then brag about it?
The whole show, I was there too when I was like not high school though. We weren't allowed to wear shorts. Oh my God. I lived in like a really repressive area, didn't I? I wasn't allowed to wear shorts. I just realized or it was like frowned upon in college, right? Yeah, that's what the deep of winter, all the guys wearing shorts. No, that's because we're stupid. That's a stupid thing. That's the whole like, I don't want to wear pants. It has nothing to do with being able to, I guess we're okay with it because we can stay on the cold long, but no, our legs don't have extra fat on them. We're just stupid. I could say that because I'm a guy. And I was there, and I could tell you the only reason I was wearing shorts was it was kind of like it was almost like a game of chicken with the guys. I think it was like an unspoken game of chicken. We were like, who's gonna start wearing pants first? Oh, I'm not. I'm fine. I'm gonna keep wearing my shorts, even though it's snowing and there's 3 feet of snow on the ground. I'm tougher than you. I'm gonna wear my shorts. I think that's what it is. There's nothing to do with. We're not uncomfortable.
Professor?
Of course we're uncomfortable. Go ahead, Jordan.
You're
You wanna
going on a
disagree with
little bit
that?
of a tangent.
That's a little bit of a tangent. I agree.
Yeah.
I'm sorry about that. It was funny. A little
I
bit.
thought it was funny. Yes.
Thank you, Taylor. I need that kind of support every once.
No, it was
Shut up,
funny.
Jordan, you're going on a tangent.
Julia.
No,
where are
it
you?
was funny. I'm just like, I'm just trying to remember what we were talking about before, so I don't have shorts and pants in my head.
Well, I need Julia to say the word lemon, but Julia apparently is not here right now.
I was thinking about it, but I was just like, you know
It's
what?
all right. You can say lemon. You can say lemon.
Okay.
I get it. I get it. All right, here we go. Incomplete dominance. Neither phenotype wins. So here's the key. This is what you can write down and memorize if you need to. The heterozygote has an intermediate phenotype. There's a third phenotype. This is what looks like blending. Okay.
But it's not.
But it's, and why not? Why isn't it actually blending?
Um,
The dominant and recessive genes can still come out.
Say again, Alexis.
The dominant and recessive genes can
Yes.
still come out.
You got it. If you remember, the blending theory, according to blending theory, you can never get back what's been blended before. Just like my coffee here, my beige coffee, I can never pour this. I can't mix and match this with 2 other beige coffees and never get black or white again. Right? Okay. You would say the reemergence of the parental traits. See a chat message that just popped up. No worries. I thought you were gonna type lemon. Okay. That's right. You went out west. Okay. Here we go. Next. Okay, epistasis. This is the idea here. One gene controls another gene. Right. And actually I'm gonna give you a specific example of this. This is a famous example of Labrador coat color retriever. The word itself literally means standing upon. And so one gene standing on the other and keeping it down. That's the idea there. We all love dogs, right? We all love dogs, right, cat people? No, it says, okay, you don't have to love dogs to understand this. It just helps. Labradors. Anybody have a lab? Any lab people? Oh, there we go. Sammy, what kind of lab you got?
He's a black lab mixed with like a pit bull a little bit.
But what color is he?
Black.
Black. Okay, we got a black lab. Anybody else? Any labs? All right, so now I'm going to teach you how the coat color works. Ready? I'm going to do this quick. I don't want to spend too much time on this. There's two genes, right? I said one gene controls another gene. The first gene makes a pigment. And that pigment is either black or brown. Okay, simple Mendelian pattern here. Black is dominant to brown. So we use the B for black, right? Don't use the B for brown. We use capital B for black, lowercase b for brown. Okay. The second gene, I want you to think of the second gene as a machine that delivers the pigments that are made by the cell to the hair follicles. It's like a taxi cab. Okay, so think of it like a taxi. It's a pigment taxi. And as long as there's a taxi there, you can deliver the payload, right? And so we use an E for this. So E is a taxi cab that works, and lowercase e is a taxi cab with flat tires. Is broken, can't do its job, right? So you can understand that the only way that you can see that phenotype is the lowercase e, lowercase e. So that's another dominant trait. I'm sorry, that's another simple dominant recessive concept. Okay, so you got two things going on here. On one hand, the cell is making a pigment that's either black or brown, and on the other, it's getting that pigment or it's not getting that pigment to the hair. All right, so you can see now, you can picture there's a hair follicle. All right, and if that hair follicle has black pigment in it, it's black. Or if it's got brown pigment in it, it's brown. Or if it doesn't have pigment, it is perceived as yellow. Okay, so a yellow lab is actually a lab that has no pigment in its hair. This is not albinism. For those of you who are like, oh, it's
This is
albino.
also how blonde hair works in like people, right?
This is
Lacking
different
pigment.
than people. Well, yeah, I'm sure we probably have pigment deposition mechanisms too, but
Okay.
and that's probably why some people have blonde hair. Actually, yeah, the blonde hair is probably the result of this kind of mutation too.
No
That's why blonde's
pigment.
rare. The lack of pigment, right? Alexa.
So epistasis like turns on a gene expression or not, kind of.
So you would say, so in this case here, what I just painted for you, you would say that gene A or gene 1 here is under epistatic control of gene 2. Okay, gene 2. So here's how it works. Here's how it works. Let's go to the slide now. Okay, so you got so fur color is either black or brown, right? Capital B, lowercase b. And then you have the pigment deposition gene. You either have the ability to deposit the pigment into the hair, so plus pigment, or you don't have the ability to do that, minus pigment. So here's the key. If you can't deposit the pigment, it's going to be yellow no matter what. It doesn't matter that the genetics give us black or brown. We can't get it there. We can't see it. All right, so if you look, for example, at capital B, lowercase c, right? So you do a dihybrid cross, the classical dihybrid cross that should give you the 9:3 to 3:1, and you don't get 9:3 to 3:1. You get a different ratio, right? So just look at this one genotype. Capital B would be black, but lowercase c means there's no pigment deposition. So they're yellow, okay? Whereas lowercase b would give us brown, but lowercase c would give us yellow, right? Yellow means the only time you see yellow is because we have no pigment. That's the key here, right? So if you look, go ahead.
I was gonna, so then like, does it always happen in, I guess that's not like a broad generalization, that like it always happens that like a gene will turn a gene on or off, or like a gene will express a gene or not? I guess, does that make sense? Like,
well, it's not always, right? So this is only when we see epistatic gene control. In other words, not every gene is controlled by other genes necessarily, right? Like Mendel's pea plant, the gene that gave rise to that one enzyme to break down chlorophyll or not, there's not another gene that controls that gene that we know of anyway. Right? Is that what your question was? Like, do we always see this? This is just something we've discovered.
I guess like the question is like epistasis is like,
Oh,
could you
yeah.
generalize
so
that
we would,
as
yeah,
like
yes.
switch?
Yeah, absolutely. Yeah. That's a good analogy. It's like a switch, right? So if you have the double lowercase e, if you're homozygous recessive for the second gene there, that's the switch being off. You know, it's kind of like walking into a room that's got a nice art exhibit where every piece of art's red, but the lights are out, so it's black. Yeah. All right. So that's epistasis, and that's Labrador color retriever, and that's the last complex mono, the unifactorial concept. Polygenic inheritance. This is just straight. I'm just going to run through these slides. More than one gene, right? So our skin pigmentation is informed by, I think there's 8 genes, and each gene is either on or off. Each gene either produces or doesn't produce a pigment, right? And so think of it like you can think of your skin tone as a bunch of those little buttons you got on your touch screen on your phone, that if you touch them, they look like this or this, right? And so you can turn them all on and you have dark skin, turn them all off, you have light skin, or you can mix and match and you have all the variations of a skin tone. Okay, so it's a spectrum. Okay. They are
So, like-
quantitative. That's the key though. I mean, right, there's only a certain number of skin tones. There's just a lot of them. And then each one you can be tan or not tan. You know, that's another layer of complexity. If I go out to the sun, for example, if I used to work at the beach when I was in high school and in college, and if I spent an entire 4 weeks in the sun, I would get like chestnut brown. But that's not my skin tone. My skin tone is whatever this pasty color is. Because I haven't been out in the sun regularly for a while now. Okay, so that's an example of where the environment can also play a role, right? All right, go ahead, Jordan.
I was gonna say like with that analogy of how you said like you can turn it on and off, it like reminds me of this video of this dude showing like this like the pigment
Lemon.
cells in like a squid and how they can turn on and off. So it's like really
Yes.
cool.
Yeah. So squid color, those things are actually the skin of a cephalopod has these special cells called chromatophores, where they have muscles around them. And if they squeeze the muscles, a little the cell basically balloons out as the color it has in it. And so that's why they can instantaneously change their skin color, because it's muscle controlled. And it's controlled by an autonomic response. They don't have conscious control over it necessarily. Right. And even more ironically, as far as we're aware, they're colorblind. And despite the fact that they're colorblind, they can still blend in with their environment perfectly, including the colors. And that's one of the great mysteries of cephalopods. Okay. Yeah, cephalopods are one of the coolest animals. They're as close as we get to aliens, because their brains, as smart as they are, and they're very smart, their brains evolved 100% independently of the mammalian brain. A complete example of convergent evolution. Their neuron complexities are absolutely different. They have a different history than ours, and yet they can still do very similar things than ours. All right, anyway, so that's that. Okay, environmental impact. Hydrangeas are an example of a flower that has a very significant environmental role. So red hydrangeas versus blue hydrangeas. And you know what? I'm gonna finally get this right. Red, one of them is low pH, one of these is high pH. Red versus blue hydrangea. I'm gonna get this right. Let's see. Okay, blue is acidic. Okay, so here, acidic, alkaline, and then so North and South America. Let's see where guide to hydrangea. Let's see. So blue, southern. Okay, so this is, hold on, this is south and this is north. Okay, so in the north we tend to have red hydrangeas because our soil is more acidic in the north, and in the south they had blue hydrangeas. They tend to be, I'm sorry, we have more alkaline soil in the north. There's more acidic soil in the south. All right, go ahead. What was that question?
Is that a sea monkey?
Is that what?
A sea monkey?
Did you say sea monkey? Is that okay?
Yeah.
That took me a second because you're talking about that. I think so. Yeah. And so yeah. So oddly, flamingos, when whatever food they're eating, the way they process their waste is in part they push it into their feathers. Just get, and that's why you have pink flamingos, because whatever they're eating, you are what you eat.
Shrimp.
What?
Shrimp?
Shrimp. Yeah, it's a type.
that what they
Well, they eat shrimp. That's a krill. No, it's not krill. That's a sea monkey apparently.
No, they eat shrimp.
Oh,
Right?
oh, you eat shrimp. Oh,
No, they do.
they do. They eat a lot of things. They eat lots of shellfish.
But that's why they're
Um,
pink.
Well, that's well,
'Cause they
no,
eat-
no. They also eat. I mean, really, most arthropods have that pigment in them. So yeah, and the shrimp actually, right? That color that you see that's the cooked color, right? In nature, they look
They're
more
very
like.
ugly and gross. Yes.
Well, yes, they are. I've had somebody tell me that can't eat shrimp because they're basically like the bugs of the sea.
Yeah.
They're just insects, but they're so good. I love shrimp.
And they're cooked. Yeah.
Yeah. All right, you see the little cute bunnies now, right? There's an enzyme that makes a pigment. The pigment's black, but the enzyme is very delicate. I call it like a snowflake enzyme, right? So if this is the enzyme, right? Even if it just gets a little warm, it just falls apart, okay? And so this is called a temperature sensitive mutation. And so if you take these bunnies and you rear them, you raise them at very cold temperatures, under 20 degrees Celsius, you will see his cute little bunny nose and his cute little bunny ears and his cute little bunny tail and his little feet, his little paws. They're all black because those are the parts of the body that are the coldest. As you know, if you're out in the winter, right, your ears are the first to get cold, and you get cold hands and your nose gets cold. That's because those are the areas that are the thinnest membranes, closest capillaries directly to the environment. Whereas if you raise that, so think of a clone of that bunny, and these are clones of each other, genetically identical rabbits, raised at above 30 degrees Celsius, you see none of that. Okay, so that phenotype is not genetic. That's due to the environment. And then lastly, identical twins, if you separate them at birth and you give them completely different diets, they will have different heights. So our heights are actually informed in part by our genes and also our diet. This is why if you ever go to a museum and you look at medieval clothing and suits of armor from the knights, you'll notice that those suits of armor look like they belong to a bunch of, like teenagers. They're like 5'2", 5'3", and that's because that was the average height back then, because we had poor diet, poor nutrition. And that's why immigrants and refugees from third world countries that were raised in environments of poor nutrition tend to be very short, but their children, once they're here and they're fed the traditional American diet, are just as tall as anybody else. Go ahead, Alexa.
I just want to quickly tangent because you did say at one point earlier on in a lecture that like we could run like dumb fast in the past, but like
Yeah.
we're short too.
Yeah.
Like where
Well,
is there correlation there?
No. We're short if we don't eat well. We're fast if we're not fast, but we used to be. Yeah, there's, I mean, I imagine early humans did not have, the best nutrition either, right? So I'm sure that in any, wild setting, humans were probably, average height 5'2", 5'1", whereas, high fructose corn syrup from birth, and here you are at 5'8" now. Yeah. All right. And then pleiotropy, one gene, many traits. There's one human disorder that I want you to know. And this you can lump this in with the human stuff from earlier, the human genetic stuff. It's called phenylketonuria, or PKU. And this is a mutation caused by one gene. It's called PAH, the PA gene, phenylalanine hydroxylase. It's an enzyme that breaks down the amino acid phenylalanine. And in normal people that don't have the mutation, right? So by the way, this diagram down here, pay attention to what I'm going to say, because when I stole this from the internet from some website on the disease. It's so confusing. I should replace this. So here's the idea. You see the blue arrow? That means it's normal. So the normal phenotype is you take the phenylalanine, you break it down, and it comes out as useful chemicals, carbon dioxide, and water, right? If you mutate it, you can't break it down, and instead phenylalanine builds up in the body as phenylpyruvic acid, and that causes the disease phenylketonuria. And phenylketonuria leads to severe mental retardation, reduced hair and skin pigmentation, and it's lethal if it's not treated. So this is one of those diseases that we can, when the kid first gets born, and we run those little barrage of tests, if they find the mutation in the PKU gene, the PA gene, then that kid can grow up so long as they have a restricted diet that has very, very low levels of phenylalanine, they can be completely normal. And this is why when you look at the back of some food products or gum packs, particularly diet stuff, aspartame is a phenylalanine derivative. And so you'll see something that says warning, pharmacokinetics contains phenylalanine. Warning, pharmacokinetics. Let me just show you a very quick picture here of what I'm talking about. You've seen this label.
Excuse me?
Yeah.
So phenylalanine is like a class of sugars then?
No, it's sweet, but the only reason why we perceive it as sweet is it triggers the same receptors that sugars do, but it is peptide based. There it is. You've seen this before. Let me show you a copy image. Go to the whiteboard. All right, you've all seen phenylketonurics contains phenylalanine. And that label is there for people with this disease. And now you know. The more you know. Rainbow. Okay.
Is that only in diet soda?
Well, no, it's anything that has aspartame. Yeah, here's your description. Aspartame is an artificial non-saccharide. Gosh, of course it's got to be in white there. It's a non-saccharide sweetener, 200 times sweeter than sucrose and commonly used as sugar, blah, blah, blah. It's a methyl ester of the aspartic acid phenylalanine dipeptide. All right, so you take aspartic acid, phenylalanine, stick them together, and then ester, make a methyl ester of that. Organic chemistry, ninjutsu, and you get something that tastes 200 times sweeter. That's why those packets only have just a little bit of it in there, because it's just so ridiculously sweet. It also has a weird aftertaste. I don't like it
I
personally.
heard that it
But
can cause cancer too, and my aunt
yeah, no, those studies, those initial studies were, I actually remember sitting in the lunchroom in college learning this. So we were debating this over, a bunch of friends were talking about it. So the initial study was taking rats and pumping their peritoneal cavities with the equivalent of 200 cans of diet soda a day, every day for months. So yeah, you know, if you're going to drink 80 cans of diet soda a day, every day for months, I don't think you would expect
And
any
being
kind
the
of
size
positive
of a
health
small
outcome.
rat.
No, well, no, it was the equivalent of.
Oh, okay.
Right, so
to
like,
say,
if
this
it's
poor
a half
rat.
kilogram, if it's a half kilogram, what we say is whatever the equivalent is for a human drinking 200 cans a day was put into him.
Okay.
Yeah,
That's
it wasn't
still a
200
lot.
cans a day put into the mouse. That's a very different scenario. Yeah. All right, so that's that. Okay. Now here we go. This is the most complicated thing you're going to learn in the genetic stuff. Morgan. So this guy here, he was a brilliant man, and he wasn't just a brilliant scientist, but really, I think Morgan's best trait was being able to identify smart people. He was really good at finding good scientists, people that he trained and hired. Eventually, he after he did his work at Columbia in New York, he moved to the West Coast and he started the experimental biology unit of the University of University of California, UC. And the people he hired there went on to win Nobel Prizes themselves. So like people that worked with Morgan, he's the #1 scientist to produce Nobel Prize winning scientists. So he just really was able to find talent when he saw it. And so you have to understand one important thing here. Biology before Morgan, and really the cusp between Mendel and Morgan, all right, everything that happened before that, biology was an experiment. Biology was an observational science, just like astronomy is today, right? We can't do experiments on stars. We can just look, right? Human genetics would be an observational science still, right? Experiments in biology were not necessarily a thing for a long time. The reason why Johns Hopkins University is so famous is because it was the very first experimental biology research university in the world. And then UCSD on the West Coast was the first on the West Coast. That's why UCSD is famous, University of San Diego. So we had people doing experiments. When Morgan started to do his work, Mendel had been rediscovered, 1901. And he set out to try to understand heredity in animals. You know, Mendel did plants. He wanted to see if the same stuff was true in animals. So he picked Drosophila melanogaster, the fruit fly, the common fruit fly. Easy to grow, cheap and easy to grow. The problem is there aren't a whole lot of naturally occurring mutations, right? So remember, Mendel could just go down to the pea man and be like, hey, can I have some green peas or some yellow peas? It was called, they call them, I'm sorry, the seed man. You can go to the seed man down in town and he can get all sorts of different peas to play with in his experiments. Morgan couldn't walk into Walmart and be like, hey, can I have some white-eyed flies? Can I have some dumpy winged flies that have white eyes? And they'll be like, please leave, sir. This is the Wendy's. So what he had to do was he had to hunt for them. And so he spent the first 2 years setting his lab up looking for naturally occurring mutations. And as the story goes, the very first white-eyed fly he found, so all fruit flies have red eyes. He found a white-eyed fly. And as the story goes, it got away. It flew away. And they were able to recapture it. But the way the anecdote goes is if they weren't able to get that white-eyed fly back and breed it, that could have set modern genetics back by a decade. You know, it took a long time to find that fly, and then it got away. Imagine being the person that it got away from after finding and looking for it for 2 years. Oh, I'm in so much trouble. What are you doing this weekend? Looking for a white-eyed fly that got away. So let's just remind ourselves here, we have our 1 to 22, right? These are called the autosomes. So technically all those recessive diseases we learned about earlier, those are autosomal recessive or autosomal dominant. All right, and then you got your sex chromosomes, right? So you can imagine if a gene is on an X chromosome, it's going to lead to interesting combinations, right? So here, let me walk you through his first experiment here. And so this is what Morgan did. I'll show you the phenotype first. We'll do this very simply here. We have found a white-eyed fly. Actually, let me do it this way. So white-eyed fly. And we're going to cross the white-eyed fly with a red-eyed fly, right? So the white-eyed fly was male. They discovered the male. So everybody, including all the females, were red. And when they did this cross, so right, this is the parental generation, the F1s were 100% red. That was exciting because they found the recessive trait, right? They didn't have a white eye and a red eye. They didn't have marbly eyes of red and white. They had, it was Mendelian, and that's what got them excited. They were able to demonstrate a Mendelian trait in an animal. All right, so then they did, by the way, it's F1, not just F, right? That's the F1. Then they did, they allowed the siblings to cross. The equivalent of, you know, the self-fertilization of the peas. These are not hermaphroditic animals. These are sexually reproducing males and females. The thing is fruit flies don't really care. You put a female and a male from the same clutch of eggs into a breeding chamber, and they'll have baby flies. Okay. The insects just don't have the same social norms. Humans do. All right. So
Does it affect their genetics though?
say again.
Does it affect their genetics? Like farm animals, like when you breed
well,
farm animals too close, like does it matter because they're flies? They're just
No,
flies?
it does. They don't live long enough where a lot of that matters, I think.
That's true.
Their lifespans are very short. So, I mean, there are some insects where their lifespan is a few hours. They're born, they get sexually mature, they have sex, they lay eggs, they die, all within a day. Yeah. All right. So, again, excitingly, right? You can think we do a red eye cross with a red eye, right? And we get the expected ratio of 3 to 1 of red to white. Monohybrid cross success. Okay, now here's where it gets interesting. There was this guy named Andrew Sturdivant. I can never do this. Andrew S. called. And he noticed under the microscope when he was looking at the insects, he noticed that the white-eyed flies were 100% male. There were no female white-eyed flies. I'm sure they did the cross again to verify it. So they just found a violation of Mendel's laws, right? Mendel did his reciprocal crosses. Well, we can't even do a reciprocal cross here. We don't have a white-eyed male or a white-eyed female, right? So this is what they hypothesized. This is what they said based on what they knew. So Drosophila, so D melanogaster has an N of 4. All right, so they have 4 chromosomes. Chromosome 1, chromosome 2, chromosome 3, and then they have either X or Y, just like we do. Okay. And so they hypothesized that the gene responsible for the white eyes must reside somewhere on the X chromosome. Right, so they mapped this out. Okay, so here would be the male. Female. Okay, so let's say that so in fruit fly genetics, the way we name the alleles with the letters we use, we don't pick the letter of the dominant trait. That's what Mendel did. What Morgan did was he picked the letter of the mutant. Right, so in this case, we would use lowercase w for white, because the white color was the mutant color. And then we would say either a little plus or nothing at all. Okay. So the females would be this, the male would be this. Okay, so a little plus denotes what we call wild type. That's the naturally occurring wild type. Okay. So when you do this cross, right, the female can only contribute a single allele, right, W plus. The male, though, can either give us the X with the mutant or a Y. And so 50-50 male female, of course, right? So the females would be W plus W. So the plus goes first, just like the capital letters go first. And then the males would be W plus. So this would be the F1s, right? 100% red. So that would be expected. Then he allowed these to cross. So let's set that cross up. So you have a red-eyed male crossed with, oops, a white-eyed female, but she is a carrier. All right, so when you map this cross out, okay, you get females that are red-eyed, females that are red-eyed, males that are red-eyed, and males that are wide-eyed. Go ahead.
So W plus, the wild type, is red eyes, and then just W is white, the mutant type, right?
Yes. There you go. And so there's your 3-to-1 red to white. Right, so this would be the F2, 3-to-1. I'm sorry. Yeah, this would be the F2 3-to-1 ratio. That's your classic monohybrid cross. But when it comes to the sex of the animal, you'll see the females are all red-eyed and the males are 50-50. So here's my question to you. This is the critical thinking. Let's say that we were in Morgan's lab. Let's say that you were, junior lab assistants in Mendel's lab, and you came up with these observations empirically. And he said to you, all right, it looks like we're on the right track, but we should do one more experiment. Let's create a wide-eyed female fly. All right, so here's what the experiment would actually yield. You're not going to see all this, right? What you're actually going to see is you're going to see this fruit fly growth chamber, which is basically a glass vial like this. It's got this kind of weird food stuff in it. That's like, it's kind of like this color. And then it has a nice foam cork in the top. All right, and so you see in there, you just see a bunch of flies crawling around. Right, and so what we do is we anesthetize them. We take a tube and we pump a bunch of carbon dioxide in there. That knocks them unconscious. And then we have this little device. It looks like a it's like a sponge almost, like a just a little cardboard pad, but the top of it is porous, and there's a little tube on the side here that we can pump carbon dioxide into. So basically carbon dioxide is able to come up here like this. And so we pour the unconscious flies onto this, and then we can look at them under the dissecting scope. And we can, you know, we use a little paintbrush so we don't hurt them, and we can just look at them. Right, that's how, and then we have a little clicker in our hands and we can count the number of flies that are white-eyed and red-eyed and all that. Okay, so you took a white-eyed male, you crossed it with a red-eyed female. That gave you 100% males and females, right? Parental F1. And then when you let those cross in the F2, you saw a 3-to-1 ratio, but you saw you saw the males were 50-50 and the females were 100% red. So what do we need to cross in the F2 to make an F3 that gives us or whatever else it is? We want a white-eyed female. Okay, so right now you have a stock of F2 flies and you have a whole bunch of them, right? In this F2, this is in a single container and there's, 60 flies in there and 25% of them are white-eyed and 75% of them are red-eyed. And all the red-eyed ones are female and half of the red-eyed ones are male. Or I should say, of the red-eyed flies, they're male and females in there. And the only ones that are in there that are white-eyed are male. So who am I crossing? Do you see the problem? It's almost like a test cross. All those red-eyed flies in there, right? If I just handed this vial to you, you bought this on Craigslist. How do you know which ones of those females are true breeding for red-eyed? You don't. And so here's what you do. This is the solution to that. You do a bunch of them. All right, so what I'm going to do here is I'm going to pull just randomly. I'm going to take 1, 2, 3, 4, 5, 6, 7, 8. Now I'm going to take 10 tubes and I'm going to put a red-eyed female into every single one of those tubes. And I'm going to put one of the white-eyed males in every single one of those tubes. Okay. Now, as a reminder, if we go back to the Punnett square here, when we look at this carefully, those red-eyed females are either this genotype or this genotype. There's 50-50. So statistically speaking, if I did this 10 times, 5 of those tubes would have one of these in it, and 5 of those tubes would have this in it. Right. So when I go back here, if the female was this, okay, so the male is always this, right? This is basically like 10 test crosses. So that would be one setup. This would be the other setup, right? And of course, you got the Y. I'm not writing, I'm leaving out the X chromosome here. But the idea here is this, let's forget the male. Let's just think of the females, that's what we're going for. All right, 50% of the time, we're going to get 100% red, right? So we would expect of these 10 test tubes, 5 of them are going to give us always 100% red females. The other side we're going to see 50-50. Raise your hand if that made sense. So make sure you're keeping up with me. Okay. Questions? All right, so Morgan's team did this experiment, and this is exactly what they saw. Okay, and this is science right here. This is pure science. Empiricism. Go ahead, Taylor.
Did they get a white eye female or were they
Yep.
all male?
Nope. So it was 50-50. Right? This is the F3 progeny. So in the F3, when they ran 10 F2 crosses, 50% of the time those crosses would yield 50-50 white-eyed, red-eyed females, and 50% of the time they would just get red-eyed females, which is exactly what was predicted by their model. Right? And so that's science. There's empirical observations, empiricism combined with rationalism, being able to reason your way through it. And so what we just came across here did two things for Morgan, and this is what put him on the map when he published this finding. He was able to show sex linkage. In other words, he was able to show that there were genetics underlying the X chromosome. But more importantly, he was able to finally identify the mystical factors of Mendel. And he was able to say that whatever the factors were that Mendel referred to were these discrete regions of a chromosome. Call them genes. I don't think Morgan came up with the phrase gene, but this is where they started talking about the gene and gene theory, the gene theory of inheritance, instead of just calling it the particulate theory, right? So this is what more, and in the publication, he would say, here's the X chromosome. And that X chromosome, somewhere on that X chromosome was a gene locus that either had a red eye variant or a white-eyed variant. Okay, so that was the first big thing Morgan did. And now what I'm about to say, this is where things get complicated. So before we even go on from here, you need to ask me questions to make sure that you can go on to the next concept. Any questions here? There's nothing, nothing here should be complicated. If you've been doing your genetics homework and you're understanding all this, there's nothing new here. We're just extending what you already understand.
So we found out from the first breeding, like from the parent into F1, that the red eye is dominant to the mutant.
Yes.
So the reason why we found the males to have white eyes more frequently than female in that F2 progeny was because they only have that one X chromosome. So it only needed one to have that. And the reason they struggled to get that with the female was because they needed two, right?
That is a perfect understanding, Taylor. You got it.
Okay.
Good. So remember when we learned Mendel, we did the monohybrid stuff, right? And then we had the whole, well, what about a second gene? The talls and the shorts and the yellows and the greens, right? What was the relationship between different genes? And so Morgan then, basically he was modeling his series of experiments after Mendel, right? So the next step here is to ask the question, what's the relationship between two genes? And so for this series of experiments, I'm going to use my PowerPoint because I actually mapped this out. So here, okay, in your textbook has a lot of very complex, nasty figures and diagrams at this point that if you read ahead and you tried to outline it all, it was probably overwhelming. Visually, it's just a nightmare, right? Because there's just so much information there. So what I'm going to do for you right now is I took the textbook's example of the dihybrid cross essentially, and I distilled it into much simpler terms that you can see on the screen here. So I'm going to walk you through this. Okay, so if you're in your studying in the future, you're going through this cross and it's not making sense, you come back to this video and watch this segment again. Okay, so I'm going to try to do this in one uninterrupted little 2-minute thing. All right. So here's the setup. We have two traits: body color, wing size. Body color is either black or gray. The natural wild type version is a gray body, and the mutant is a black body. So we're going to use the B that represents black, right? And so B plus would be gray, and B would be black. All right. So this would be the natural version that they find in nature. This would be the mutant. Okay. Wing size. You can have normal size wings, or you can have vestigial wings. Vestigial means it lost its original function. Basically, they're miniaturized. So these flies can't fly, which begs the question, what do we call them? Let's call them walks. Okay, so we have flies and walks. All right, so vestigial wings, VG. That's that's the stand-in for wing size, right? So VG positive means you have normal sized wings, and VG means you just have little tiny wings. You can't fly, right? So what you can think of here is we're going to take, right? So a wild type fly would be, right, B plus B plus, VG plus VG plus, right? So that's their generic flight find out in nature, and then the mutant would have to be this, because these are all recessive genes. That's important. Okay, so he didn't just find these in nature. He bred these things over years. This took a while to establish, right? And so at one point in time, the Columbian Fly Lab was famous. It was a world famous genetics lab. And you can imagine walking into a room filled with cupboards, filled with little vials of flies that all had identifying marks on it that basically had these kinds of labels on them. All right, so any researcher can come in and mix and match and do crosses. All right. This is also, you can see, well, I'll just leave it at that. So this is the parentals, right? Those are the parentals. We're going to cross them. And we're going to get what we expect, gray-bodied, normal winged fly. And what we're going to do then is what's called it's a test cross, right? We're gonna take the dihybrid here, right? And we're gonna cross that. It's also called a back cross. When you take an F1, you cross it back to a parent. Because remember, again, flies don't care. So we're gonna take the original mutant, right? The homozygous recessive for both traits. And we're gonna cross that with the F1. That's a test cross, but it's also called a back cross because we're pulling it from the previous generation. So in other words, we're going to do this cross. We're going to do the B plus B, VG plus VG crossed with the homozygous recessive. Right? And the reason for that is the number of unique alleles this one can make is simple, right? It's just B, VG. That's the gamete phenotype or genotype, right? It's simple. There's only one. Whereas this one's going to have all four, right? Remember the FOIL method. So our Punnett square here, really our Punnett rectangle, is going to be a 4x1, right? All four of the different combinations here. No, here I have it mapped out over here. Look at this. Okay, so here are the gametes crossed to the test cross. Okay. And so let's reason our way through the possibilities. Okay, we would expect if fruit flies behaved like pea plants, we would expect to see one of each of the phenotypes. So let's just refresh our memories. What are the phenotypes? We got gray body, black body, we got normal wings and vestigial wings. So we would expect to see right here, what would the phenotype of that one be? Let's see if we're all on the same page. What's the phenotype of that fly?
Like true wild type.
Exactly, gray body, normal wing. All right, what about this fly?
Black body, normal wing. Mutant.
right? It's homozygous recessive, right?
Yeah.
So that'd be vestigial winged. Right, that's so the mutant is vestigial, and that would give us the mutant phenotype. So this would be 100% mutant. What about this one?
Vestigial wings and was it black body or is it gray body?
For what? Wild type or mutant? Remember that the letter is taken from the mutation. So the B tells us that black is the mutant and therefore gray is wild type. So is that gray or black?
It's gray.
Good.
It's
Okay,
gray
so
and
we-
vestigial wings.
You got it. Gray vestigial. And then what's this one? Be the opposite, right? It'd be black and normal. All right, so here's the key. Here's one of the little secrets to all this. Notice that for the ones that have the recombinant phenotypes, right? That you get one parental and one recombinant, or one recombinant and one parental, right? So you're either the wild type for one trait and mutant for the other, or you're mutant for the one trait and recombinant for the other, right? So the idea here, right? The idea here is that these two phenotypes, these two genotypes, we're going to call these the recombinant ones, right? Because again, they have one trait from mom and one trait from dad. They don't look like either mom or dad. They're a combination of both, whereas these are the parentals. Okay, that's I've introduced that concept a couple times now. This is where the payoff is. This is where it's very important to understand this now. All right, so does that make sense? The idea of the parental phenotypes, the parental genotypes, the recombinant ones. Okay, good. So when they map this out conceptually before doing the cross, they would expect to see even representation across the board of all of them if the genes for the two traits are on different chromosomes, right? So let's just think about it. Here's chromosome 1, chromosome 2, chromosome 3, chromosome X or Y. And let's say that the trait here, chromosome 1, 2, 3, and then let's say, X or Y, doesn't matter. Okay, and then we have the body color. Okay, so B. Let's say that the gene for body color is here, and the gene for vestigialness is over here. Right? Can we all agree that if those genes are where those are, when we go through meiosis and crossing over and all that jazz, we're going to see independent assortment, right? We're going to be able to produce all the various combinations, right? So here's the diploid. Let's say that this is the heterozygote, right? So that's the visual representation of the genome of the dihybrid. And so you can see when we go through meiosis there, right? We can get the B allele with the VG allele. We can get the B plus allele with the VG allele, or we can get the B allele with the VG plus, or we can get the B plus with the VG plus, right? All 4 possible combinations are there because of independent assortment, right? You guys with me? Okay, but it's the same idea as the tall shorts and the yellow greens, right? We're sitting here assuming that the genes for each of those traits are on different chromosomes. If they're on different chromosomes, we're going to have we're going to have independent assortment occur so that all possible combinations of gametes can be made, right? Remember, so each one of these is a gamete genotype. Okay, ask me questions here because it's just going to get more complicated. Alexa, you look confused and that worries me.
So if they're on the same chromosome, you wouldn't see as much variation in the gametes.
Yeah,
because they'd be
right.
stuck
But before
together.
you don't jump to that yet. I want to make sure you understand that if they're on different chromosomes, this is what we've learned so far. This would be normal, right? So does this make sense to you?
So okay, so just so like I understand this right. So like since they are on different chromosomes, you're able to get that variation because they can like independently assort, like you were saying.
Okay, yes. So you get that. Okay, so now we'll do what you just said. So what if I did this? What if I took that gene from chromosome 3 and I moved it over here? Now am I going to get independent assortment? No. I'm always going to get either BVG or B plus VG plus, right? I can't get B VG plus or VG B plus VG. I can't get those because they're on the same chromosome. I'm going to get this. It's all or nothing. Forget crossing over. Forget crossing over. Just think of the fact that they're on the same chromosome right now. Okay, linkage. We call this linkage. Okay, so if the genes are on the same chromosome, we would expect this ratio. Okay, forget this. We would expect if these two genes for body color and for wing size, if they were on the same chromosome, we would expect to never see those recombinant phenotypes. They would just be 0. We would do the cross, we would see the F2s, and we would see only gray-bodied normal or black-bodied vestigial, and that's it, just like mom and dad. We would never, so in a 1-to-1 ratio, 50-50. Okay, so before we forget crossing over. They didn't know what that was back then. They barely understood meiosis, right? So these were the only 2 worlds of possibilities for the researchers doing this cross. Okay, their hypothesis led to a model prediction. And so when they did the cross, they didn't get either of those. These numbers down here were the original numbers taken from the original paper they published. 965 flies were counted that looked gray-bodied and normal, just like mom. 944 were black-bodied and vestigial, just like dad. A little fewer, 944. 206 had mom's gray body and dad's vestigial wings, and 185 had dad's black body but mom's normal wings. And they cried and went home and took a long weekend. Because when you do an experiment in the lab, when you're a researcher, whether you're a grad student or a senior investigator, every experiment you're doing, you already have mapped out the possible outcomes. That's how you do science. And when you get results that don't match anything you predicted, you get really upset because that means you probably made a mistake somewhere. That's why you have to repeat the experiment at least 3 times. And if you get, that's the rule of thumb. If you do an experiment and it gives you results that you're not expecting, you have to repeat. Actually, what you're supposed to do is repeat all experiments 3 times, no matter what, even if you get the right results. Otherwise, you get something called confirmation bias, right? This looks right, it's good enough, let's publish it, right? When you made a mistake and it looks, right? You can still make mistakes and it just gives you a nice outcome. Still mistakes though. So anyway, they do this a bunch of times and they keep getting these same results, right? So now, after they got over being upset, they were able to look at this with a cold, a new set of eyes. Or maybe, so what you do actually in the lab, when you get in a result that doesn't make sense to you, you share your data with other people and you ask them if they have any insight. Because they don't have any skin in the game, right? They have an objective view of it. They can probably see something that you're missing. And so ready, watch this. What if I said to you, well, okay, those numbers are all wrong. Like these are clearly not 0. They exist. But these numbers are not evenly divided throughout them, 1 to 1 to 1 to 1, right? But if I told you to round these numbers to the closest 1,000, round up, I'm sorry, just round to the nearest 1,000. This would be 1,000, right? 965 rounded to the nearest 1,000 is 1,000, and this is 1,000. And then 206 rounded to the nearest 1,000 is 0. And so is this, right? So in other words, these results kind of look like these results, but these are problematic. Okay, so this is what they thought in their head. Let's say hypothetically that on occasion during meiosis, the homologs can occasionally cross over. All right, watch this. So here we are. Here is a map of a chromosome. Okay. And those two clips represent the two genes that we're talking about on the same chromosome. So linkage, we've established linkage here. If crossing over occurs randomly and it happens to occur in between those two genes, we're going to see the recombinant phenotypes. All right, so let's draw this out for a second so we can remember what this looks like. Okay, so here we go. So here's chromosome, and here's the gene. Let's keep this simple. Let's do the capital and lowercase a that you guys are more familiar with here, so you can remember the concept. Right? If crossing over occurs anywhere between them, that will result in these two new recombinant chromosomes, right? Right, remember we're going to do this. So that's going to give us the A and the lowercase a. So here, this one here would be represented here. This one here would be represented here, right? You see that. If crossing over is completely random, and so here's where things get both amazing and important. This is the key to today's lecture. Ready? The closer those genes are together, the lower the percent chance that crossing over is going to occur between them, right? Whereas if the genes happened to be all the way on these ends here, can we agree that those genes are always going to have crossing over occur between them practically? Right? So again, just pick a random spot on this meter stick, on this ruler. If that random spot occurs somewhere in the middle here, we're going to see crossing over occur, right? Okay, you look confused, Alexa.
So I guess like the more space that you have between these linked genes, the like higher probability that crossing over will occur because you have a bigger space for it to happen.
Yeah, exactly.
Is that like the, okay.
Perfect. That is a perfect way to think of it. That is literally what happens. Good. Okay, so now we can agree that if crossing over does occur, we're going to see the recombinant offspring. But we're not going to always see the recombinant offspring, right? When we do, so in other words, the frequency, or rather, when we count all the offspring, the proportion of offspring that give us a recombinant phenotype correlates with how close those two genes are on the chromosome. The closer they are on the chromosome, the less frequently we will see recombinant offspring. The further apart they are, the more common we will see them. Does that make sense?
Yes.
Okay. Taylor, does that make sense? Okay, good. You're the only face. Zach, does that make sense? Okay. Brandon, I can kind of see your head. I can see a nod if you said, yes, that makes sense. All right, good. Thumbs up is good. All right, good. April, I can see you too. Okay, is anybody, because here's the thing. Here's my worry. To some of you, this still doesn't make sense, but you're not going to tell me and you're going to get lost now. Because what comes next is we're going to map chromosomes. So speak now or forever hold your peace. Peas. Just the yellow ones. That wasn't funny. Man, tough crowd today. All right. Ready? So those numbers there. Here's what I want you to do. I want you to calculate the percentage of the offspring of this cross that were recombinant. How would you don't have to actually do it. How would you do that? How would you do that mathematical calculation? What percentage of these offspring are recombinant?
You'd have to find the total and then add together the recombinants and then divide them by that total and then multiply them by 100.
Perfect. All right, we would add 206. So go ahead, ask your phone to do this for you. Let's get a number here. So I'm going to say 206 plus 185, that gives me 391. Okay, so we would say there's three, right? We had to count one of each, right? We had to count both of those. That's one of the secrets when you're doing these problems on the test. You have to count both of those up for every black bodied normal you get. You have to get a gray bodied vestigial, right? It's always going to be both of those are both going to be present. So you have to add those together. Okay. And now, so we get 391. Now let's add 965 plus 944. That gives us 1,909, right? So plus 391. So that gives us 2,300 flies. And so if we do 391 over 200 on 3,000, we get 17.17, which is 17%. Wow, nice and even, right? We got lucky there. No decimal points. All right, so ready? I can say this out loud now. When I take a normal gray-bodied, normal sized winged fly, and I cross that to a black-bodied vestigial winged fly, there is a 17% chance that I'm going to get a recombinant offspring, right? So when they do the original work, I'm sure somebody's like, why is it 17%? Is that some kind of magical number like 3 to 1 or 9 to 3 to 3 to 1, right? Right, that's got to be there. Like somebody's got to ask, like, is there something special about 70%? The answer is no. That 17% corresponds to just how far apart these two things are. But here's what they did. They then created an entire collection of true breeding dihybrid flies and performed an insane number of crosses. This is where the Columbian Fly Lab really got its fame from. And I'll show you this picture here. This is not from your textbook. I stole this from my old textbook because this is the best slide to teach this on. It looks overwhelming at first, so let me walk you through this. On the top here correspond to all the phenotypes that are wild type. You have a gene that gives us long antenna. Okay, and so if you look at the bottom here, the mutant of the long antenna will be short antenna, right? So you can see each of these is a Mendelian trait in and of itself. So you have long antenna or short antenna, long wings or dumpy wings, long legs or short legs, gray body, black body. And so there's a couple eye colors, right? There's red eyes and purple eyes. There's long wings and vestigial wings or short wings. There's your walk, right? You can see this poor guy here, he can't fly. Straight wings or curved wings, red eyes and brown eyes. And by the way, you see dumpy wings? That's the beginning of me telling you that Drosophila geneticists come up with all sorts of weird, ridiculous names. And the reason for that is they were dealing with so many phenotypes, they had to start getting creative in their nomenclature. And they carry that on to this day. All right, so now ready? We're gonna take, so let's call these, let's just give these letters here so we can actually see how this is gonna work. Okay, so let's blend our understanding of what we already understand with Mendel and apply this here. All right, so what you can do is just pick and choose two random letters. Let's just use A and B for now. All right, so we're gonna do this cross. We're gonna take a dihybrid for those two genes, and we're gonna cross it. We'll do a test cross. We're gonna cross it with that. Okay. You can imagine a cross where you take C and G and cross that with CG. Right, so any two of these, you can do a test cross with a dihybrid. And when you do those crosses and you count the number of recombinants, not only are they always different from each other, but they're always the same when you repeat the experiment. This told Morgan that genes reside in specific locations on chromosomes. He was not only able to say, hey, Mendel's factors are real, but we can tell you where they are. We can map chromosomes. And so what they do is they invented this technique where for every 1% chance of recombination, they would call that a map unit. Okay, and so you see these numbers on the middle here now. From this gene to this gene, So when I did this cross, I got a 13% chance recombination. And so I'm going to say those two genes are 13 map units apart. All right. If I, in theory, did another cross where I took the A and the C gene and I crossed those together, I would get a 31% chance. If I did the B and the C, I would get a 18% chance. In other words, you see these numbers here? This is the map of the chromosome. Here's where the gene A is, here's where gene B is, and here where gene 3 is. So in other words, this distance here, that would be the distance from A to B. This distance would be from A to C. This distance would be from B to C. So you can think about getting a set of data, right, where I tell you the percent recombination frequencies, and you can map the chromosome.
Is that what the 17% was that we found on the previous slide?
Exactly. The 17% was 17 map units. In other words, we would say that these two genes are 17 map units apart from each other. Now, a map unit is a relative term, right? That's not a number of base pairs. It's not an inch. It's a wishy-washy thing. It's unitless, right? Well, it's not unitless, it's percent recombination. So it's the equivalent of me saying this, right? Let's say that there was a map of a neighborhood, or let's say that there was a road, right? And you had a bunch of friends that lived on that road. And I told you that, so I have my friends, I got my friends, Raph, Greg, and Jason, okay, and Andrew. And I told you that Andrew lives twice as far from Raph as he does Jason. And Jason lives twice as far from Raph as Greg. And Greg and Andrew live right? So the idea is like, well, so how many miles away is Andrew? Like, I have no idea how many miles that is. I can just tell you I'm twice as far from Jason as Raph, right? So you can say, oh, okay, so based on the logic of it, you would be able, to map it like something like that. Okay, so these are mapping problems that you'll get in the homework. Okay, now for the test, I'm not going to make you do mapping problems. Okay, that would be something you will do when you take genetics. You will relearn all this stuff in your level 2 biology class when you do genetics. And by the time you get there, it'll make more sense and you'll actually have to do mapping problems. Even if they're still teaching that. I don't even know if they're still making you do mapping problems. I think they are. Okay, so that's the basics of linkage. So this is from your book. This is the raw example from your book. And now you can pull this apart. Again, there's a lot of stuff on the screen, but if you take your time and you walk through it slowly, it'll all make perfect sense. Okay. And since it's going to be on the test anyway, map all this through with meiosis. Go through the generation of the gametes using meiosis with this. So draw meiosis out with that chromosome. And you can see, and put some little crossing overs in there and do independent assortment, and it all fits together perfectly. Because it turns out this is all true. Go ahead, Alexa. You have that look of like, is this too much too fast?
It's a little fast.
Yeah, I know. But again, this is the once over. This is, it's not like, okay, now everybody, let's take a test. No, you're going to have a few days to chew on this now. Okay. So that's crossing over.
What is the chapters that are on the test?
Let me see if I can do this. So this is 15, 14. So the little bit of wait, do we do a little bit of 11? Oh my gosh, I'm having flashbacks from the last test. Yeah, a little bit of 11 to 15. But again, remember that most of especially chapter 13 is a short chapter. Really, chapters 12 and 13 should have been one chapter. That's cell division. Yeah. Yeah, so 11 through 15. And also remember that we only did parts of 11. We only did 11.3. Okay. All right. You can watch this animation on your own. Leo combinations. Okay, so linkage map. There he is. There's Andrew Sturtevant. This is him in I think this is like 1953 or something. So now he's an old man in the 50s, right? He was the junior postdoc in Morgan's lab. You can see behind him. So this is his lab. This is him many years later. This isn't the famous Columbia Fly Lab. But you can see each one of these little vials here, and you can see this is how you grow Drosophila. And you can see they all have little labels on it, you know. So, and you know, he's smoking his pipe in the lab because it's the 50s, and you know, just smoke your pipe in the lab. This is right here. This is most likely filled with ether, and that's where you dump flies when you're done with them. So this will probably collect, if I had to guess. You know, when I took genetics when I was an undergrad, the lab component of it was a fly lab, and the guy that ran it was just a hot mess. All our flies died every week. I don't think any of our I think one of our experiments worked. The first day of class, he's like, "This will be an easy lab, and you won't have to hand in a lab report until I hand you back your first lab report," which I'm still waiting on. Never got that. Yeah. Anyway, I got stories. I'll save those for some other time. While we're chatting, ask me about my genetics lab experience. I'll tell you how to act accordingly if you ever have a professor that terrible. Okay. So this is from your book. You can see the actual linkage maps here. So again, those numbers represent the % chance that crossing over will occur between these areas, right? As for how you get numbers greater than 100%, I still don't have a good answer for you. I think it's a statistical reason. It's statistics. It's, you know, that % error within doing actual experiments versus theoretical. Okay. And oh, wow, I did it. I think I did that as a record for me. It's 12 o'clock and I got through this. I need to go buy a cake to celebrate. This is usually the part where I'm like so stressed to get everything done for the test, and we did it. Okay, so you guys we can do practice problems. We can go through and practice. We can relearn something. You can, if you want, if you kind of are tapped out, I'll say go take a nap and then go study. You got a test on Friday. I'm going to see you Friday again. So, but I'll tell you what, I can pull up the homework if you want. We can pull up the homework and go through some homework problems. That's one idea. Another idea is I can just pull up some practice problems and we can work through some. What do you want to do?
I like the practice problems just because the homework isn't as close to the test.
It's not as what?
It's not like the practice problems will probably be closer to the test questions than the homework questions would be.
Okay, so here, let me see. I want to go into my, what do they call it? The instructor resources. Where are they? Instructor resources. I'm sure that I can pull out, they have like clicker questions. I think they even have like a Jeopardy-like game. Maybe we could do that. Let's see. Resources by chapter. This is fifth. Oh gosh, I'm in the microbiology book. We're not doing theory of disease. Okay, on a completely different topic. But yeah, so again, if you leave now, I won't be offended. If you're tired and you don't want to work, that's not an excuse. Suck it up. Unless you're sick, then go take a nap. All right, let's see. Instructor resources, item library. No, not item library. Instructor resource. Come on, where are they? There we go. All right, dig in here. They have all this stuff hiding behind so many firewall stuff or whatever. Resources by chapter. Here we go. 15 art and photo. Okay, clicker questions. I'll download those.
I have a question. Do you
Yeah,
want
go
to take a small break before we do this?
Oh yeah, we could do that.
Does anybody else need that? Because I would just
Oh,
want
I'm
to
sure
get.
everybody needs that.
Absolutely.
Yeah.
Yeah,
There's,
raise
like,
your hand if you don't need a break. Oh. Taylor, the hyperfunctional one. I'm just jealous. I wish I had that kind of energy. I say with my wife, what's she doing the other day? Like,
Energy.
she's like, it's more than energy. It's like this drive to just get done as many tasks as possible in a given
Yeah.
time period.
Yeah. I mean, I have a 3-hour lecture after this 3-hour lecture.
I
So,
would
like,
simply die.
well, I'm
Right.
used to it at this point, you know, like I've been doing it all semester. So it's like,
Right, you're just acclimated to
it's like go, go, go.
it.
I
All
literally
right.
take naps after all my classes.
Oh no, there's no more time for sleep. Sleep is over for today.
So, I'll sleep when I'm dead.
Literally. Like I still don't understand how I get stuff done even though I sleep so much. My sleep schedule is so messed
Well,
up.
so
it's-
Jordan, you sound kind of like me, and that is like what I have spurts of hyperfunctionality. I think it goes along with having ADD. I'm not saying you do. I'm just for me. The
I might. I don't even know at this point.
yeah. Well, there's so many undiagnosed cases of it, and so one of the hallmarks of ADD is going into flow very easily, being hyper focused.
Because like I can literally sleep like a whole day and then I can like sleep for an hour and then I'll be awake like perfectly fine. It's so weird.
I hear you. I have a similar experience. Can you guys see this? The chromosomal basis of inheritance? All right.
Yes.
So what this is going to be, I'll leave the first one here. This is it's practice multiple choice questions. So not all these are going to be practice problems for genetics, but so I'll stop here for now. We'll do a 10-minute break. Right. So that sound like a good number of minutes? 10?
Yes. I also have a quick question.
All right. So hold on, Jordan. So, okay, everybody can go 10 minutes. Hey Siri, set a timer for 10 minutes. Okay. Okay, go ahead, Jordan.
Is the lab due Sunday or is it due Thursday?
So I always have the labs that are due before the test Thursday because if you see that, that's because those labs will help you understand the material better.
Okay.
So
I
I
just
would
wanted
say-
to double check.
Yep. And if you can't get them done because you've committed to studying this in a different way and that just doesn't work for you, I'll move it to Sunday for those people. I've done that once or twice, but most people get them done. These genetics labs, when we're in person, we don't have really a good genetics lab. So Labster's really got some good stuff in there, hopefully. I haven't walked through the genetics labs yet, but I'm assuming they're just as good as the others. But the Labster's good in general. Are they helpful and good?
All
They're
the
hit and
lobsters
miss,
are
yeah.
just weird.
Would you say
Some of them are actually helpful, then others are just like
say that again? I missed. what
They're
you said.
weird.
They're weird.
The situations that they put you in. They're like, you're a CEO coming in, doing a meeting, and I'm like, okay.
You have a meeting?
Yeah,
In
there's
the-
like one where you had to come in and then like try and figure out something.
Okay, so it's almost like they put you in situations that some weird committee thought students would be like comfortable with, and it turns out they got those wrong.
Yeah, they're just weird.
So
Those scenarios
besides
are
the
so
weird, would you say, Alexa?
I was gonna say those scenarios are so funny because it's like present your findings to a group, and like if you get the multiple choice answer wrong, even though like you're the one who's expressing like this information, they're like, I don't think that really is a necessary thing to say. Like it's so funny.
That's so funny. So wait, they have responses if you get them wrong like that?
When you're presenting information as like a scientist.
Man,
yeah,
that's-
sometimes it's really funny. I think the funniest one was like when the AIs just like have the blank expressions on their faces. Like the one lab, there was this dude who had his like the nose piece of his glasses were like up towards his eyes and not resting on his nose. And I'm like, what is wrong with the simulation?
It's funny.
like, and he didn't even have like the parts of the glasses that go over his ears. So how the glasses were staying on his face was beyond me.
Is there any way we could see the vole? And see how he's doing?
What'd you say?
Can we see the vole?
Oh, yeah, sure. I'll go
Bring
get
some
him.
wholesomeness into the break.
I'll be right back. Yeah, I think he's so I find his motor skills are getting worse and worse, I think. But he is so acclimated. Watch this. I can put him on his little back now. You can't really see him. There he
Can
goes.
you stop sharing the screen so we can get the full view?
Say again?
Can you stop sharing the screen so we can see him in like full screen?
Little face. There he is.
If you double click on like
Oh,
this page, you
oh
can
yeah. Let me stop sharing. There we go. Is that better? All right. And here, I'll make myself bigger too. So speaker view. And there we go. Right, so there he is. And he's so comfortable that I can put him on his back now. And he doesn't try to roll over anymore.
He loves you. That's amazing. Look how you did
Yeah,
that.
he really does. Oh, he does. He'll like lick here. Hold on. Although again, what?
He looks fat.
He's really not though. And he just pooped on my. Yeah, he's out of his element here, but he seems to be more and more, or less and less energetic. So I'm just getting a little worried about him. There he goes. He's licking me.
Do your kids hold him?
His kid. There he's. Oh, now he's doing his like joyful pumping. He like, it's almost like he's like ha ha ha. He just starts puffing. So there he is.
Do your kids hold him?
They'll pet him, but I'm still worried that my wife can hold him, but he doesn't. It's like I think he smells us. I think he knows us by smell, but his teeth too seem really yellow. I'm not brushing them. My worry is like, I know rodent teeth grow too much. And so I don't know if I'm giving him the proper stuff in his cage to keep his teeth short. There he is. Hey, buddy.
Just like throw a branch in there or something, right?
What'd you say?
You can chew on that. If you like throw like wood in there, I know like bunnies do that.
Yeah, and he does
wood.
that. He chews on wood a lot. So I'm guessing he's doing what he's supposed to be doing. But he's really happy and spoiled. And let's see here. I'll start licking me here. Oh, there he goes. So he's not biting me. He's licking. You see the little tongue, his cute little tongue. And he's purring right now. You can't hear him. You can hardly hear him when he purrs, but like his whole head vibrates. You can just feel it. It's like a little constant vibration. So there, oh, he's licking me now. That's him grooming me. Yeah, he's unexpectedly cute. Oh, he just bit me. Oh, what was that? Well, I mean,
he's unexpectedly.
That was the first time.
cute
That
and
was the first time he's
whites
done that. But he doesn't seem to know what he's doing. Oh,
That he literally just.
Oh, you
pulled
know,
a
he was
cat on
going.
you.
He was eating his poop. There he goes. There's 2 little pieces of poop there. There. Go ahead. Eat your.
That's even better.
No, they're supposed to do that supposedly. So, all right. Yeah, so that's that. And it's interesting. He didn't bite me to draw, like I've gotten bit before by like a hamster. They like, yeah, right. That what he just did was he nipped. So that was like an exploratory bite.
Oh
right?
my God. I've seen videos
Yeah,
of
see,
like
he's hungry. That's what he is. He's hungry right now. That's what he's doing. Let me go dump some blueberries into his cage and be done with this. So there he is. He still doesn't have a name. because again, he's probably gonna die soon. This might be the last time you see him alive. Bye. My wife wants to call him Blueberry. Not very imaginative. He eats blueberries. I like Morty, Voldemort, Morty, and Morty, Rick and Morty. Come on, Morty, let's go.
I just like I love the videos of like when hamsters bite their owners and like their owners end up eating their hamsters across the room accidentally. You know what I'm talking about? It's so dark to say, but it's like kind of funny.
I remember I had a hamster in grade school and he had like one of those like 2-tiered cages. So he had like a little tube that went up to his little like nest up top. And like I'm like 8 years old, so I'm like my little fingers like in front of his face while he's like in the tube and he bit me and I like flung my hand up and I like sprayed blood. It was nasty.
Oh my god, I had a hamster
That's
where the teeth were super
amazing.
long. I didn't know that it could even grow, but I was like, holy crap. And it like chewed it down, obviously, but they were like long.
I had a fish tank with like a carnival fish that lasted like 7 years. And we decided to take my brother's beta fish. I was like, okay, because that's what my friend did. He took his beta fish and actually put it in his bigger tank, and the beta fish didn't like attack any other fish. It was just like, it was chill. So I was like, okay, let me try that. So I put my brother's beta fish in in my big tank, and it just chilled by the filter. I was like, okay. Next thing you know, I'm like, where the hell's the beta fish? We still have no clue what happened to the beta fish, but I assume my 7-year-old fish ate the beta fish. And just like either that or the beta fish jumped out. We never found its corpse, but yeah, because like the way that the top of the tank was, there was a little like a small opening that was exposed where the filter would go in. So like the beta fish was small enough that it could jump out if it wanted to. But like it wouldn't be the first time I've had a fish try to commit suicide. I love how Alexa's like super surprised.
I just like you don't hear that. Like that wasn't the first time that I had a fish try to commit suicide. Like Jesus.
Literally, like the 7-year-old fish jumped out of the tank too once. I forget because I think it was before we got like the better top for the tank, and I think I had the top off because I was cleaning the tank or something like that. And next thing I know, I'm like, where the hell are my 2 fish? And then like I had the tiniest fish that I named Tiny Tim. I ironically ended up dying because I couldn't find him fast enough. And then the other one was behind the tank, and we managed to save him. That was the 7-year-old one. It was a hot mess. Like owning fish is like a roller coaster. Even though you can't pet them and give them love like a dog, they're still like just like crazy.
Yeah, I had a couple beta fish. They're cool. They're really pretty.
I have so many bad beta stories. I really shouldn't, but I do. For Christmas one year, my brother got his girlfriend a beta fish, or 2 beta fish, and I got to pick which one to give her for Christmas. And I wake up Christmas morning, and I'm like, okay. So I go upstairs to go retrieve the fish. Well, unfortunately, the fish was dead. So I came downstairs like I was apparently ghost white, and I was like, the fish died. So I had to give her the one I wanted to keep. And I think she named it Cinnamon.
Cute.
April, I want to give your dog some love too.
She's so sweet. Oh my goodness. She's finally settling into the house because it's been about 2 months now, or 2.5.
Oh my God, you just got her like recently?
Yeah, I got
Sapphire,
her.
this isn't about you.
I got her in like end of March, or actually no, not end of March. I mean end of February.
I love her. I die for her.
She's a cutie, and she's such a sweetheart.
How many ferrets do you have? Do you just have one or do you have multiple?
I have one, but every time I go to get him food, they always have ferrets for sale, and they're always like the babies, and it's so hard not to buy another one, but I know I cannot have another one.
Honestly, I'd like buy like 4 ferrets. They're so cute. And they're literally insane. Like I love watching the TikToks of ferrets and like it looks like they're having a mental breakdown every time they run and it's so funny.
Yeah, it's so funny when they run because they like hop and they'll arch their back up, and he likes to chase me around the room and I'll chase him too. It's like this little game that we play.
I love that. My husky's literally broken, so like I feel like my husky's not actually a husky. Like I sit there and I watch videos on TikTok of like just huskies like just screaming for no reason, and she does not make any noise like at all.
Well, you got lucky with that then, because I have for work, we have 3 huskies that come in, and as soon as we get them in the tub, they're screaming the entire time, and it's so bad.
Watch videos of them because they're just so over dramatic. Like, don't get me wrong, trying to bathe her was like trying to pull teeth. Like when we tried to bathe her, back before we got a groomer, literally our arms were covered in scratches and were all red just because she did not want to be in the tub at all. And like
Yeah,
she would
it's weird.
whimper a little bit.
They don't
But
like water. I don't get it.
No, but that's the weird thing. She loves to swim. When we went
Yep.
on a houseboat for vacation, she was in the water and she was swimming no issue. And she goes, like, when we go to the beach, she'll go in the water and she'll get all wet, but she doesn't like being bathed. It makes no sense to me whatsoever.
I feel like it's like if the water's on their terms, they're fine with it, but once it's forced on them, they're like, "Oh no, I'm gonna die right now. Like you need to stop." So
Literally, like I didn't even put her in the tub the one time because I think she rolled around and pooped the one time, so I had to wash her off. And I didn't even put her in the tub. I just grabbed a bucket, grabbed the soap, and she's like literally huddled against the door, and I kind of have to like pull her away in order to get her other side to wash her, and she's just like not having it. I'm like, dude, I'm not gonna kill you. It's water. And she's just like weird because she'll only talk when she wants food or water or needs to go out. That's the only time she'll ever talk. But like she can sit in front of our glass door, our storm door, and the mailman can come right up to the door and she won't even move a muscle. She's like the worst guard dog ever.
You're in the middle of a story.
welcome back, Professor.
We've just been talking about animals.
All right.
Wait. I want to show this husky video I found.
Yeah, I think it's because I separated my hearing aid from the Bluetooth. So just gotta find it again. And come on.
Never mind, I lost the video.
There
I'm
we go.
sad
Yeah,
now.
I can hear you. Okay. Huh. Today's a big day for the cryptocurrency market. Coinbase, which is the premier exchange, went public, and so today's kind of a make or break day for Bitcoin and all that. So the prices are all over the place. It's a very interesting day.
Wait, can I show a husky video I found?
Wait, what? Did you just say can? Yeah, sure.
Can I show a husky video I found?
Yeah, go ahead, share it, and then
It's
and then
so
we'll
funny.
do genetics.
I love it so much because she's playing with the dog and she makes a noise and the dog just looks at her and screams. It's so funny.
I love when dogs sing along. My dog Buttercup, when she's outside hanging out and there's either an ambulance or a fire or a police siren, which isn't that common around here, she'll just start howling with it, like really loud, like a wolf. It's just like it brings out her inner wolf.
I was gonna say they
Just so-
probably think it's like another like dog howling just because of the frequency of it.
Yeah, that makes sense. I always thought she did it because it hurt her ears and like she would do that to like drown it out. But then I realized that was probably a social cue, as you're
Yeah,
saying.
like all huskies howl except mine. Mine doesn't scream. Mine doesn't
Except
howl.
yours.
She's like broken.
He's just different. All right, you guys can see the question here, right? Let's
No.
go through some of these. Okay, so let's see. How did improvement
see
in
it.
micro? But yeah.
We don't-
Can't see it.
Just you.
Oh, you can't see it. That's right, because I have to share again. There we go. All right, so how did
Okay.
improvement of microscopy techniques in the late 1800s set the stage for the emergence of modern genetics? So what do we think here? It reveals new and unanticipated features of Mendel's pea plant varieties. It allowed the study of meiosis and mitosis, revealing parallels between behaviors of the Mendelian concept of the gene and the movement or pairing of chromosomes. That's probably the right answer, right? It allowed scientists to see the nucleotide sequence of DNA. No, you can't see that under the microscope. It led to the discovery of mitochondria. While that's true, that's not related to the emergence of genetics, right? It showed genes functioning to direct the formation of enzymes. Well, not at all. So I'm going to guess the answer is B here. You guys agree? That's yeah. All right. Ah. Oh, it was B. That's surprising. Okay. All right, here we go. Morgan and his colleagues worked out a set of symbols to represent fly genotypes. Which of the following is representative? We just did this. So let's see. Were you paying attention in class today? What do you think the answer is? Type it in chat if you want, instead of calling it out loud. That way, if you don't want to know what everybody else thinks, don't look at chat yet. Yeah, okay. So that's the only one that makes sense. Yeah, so of course the answer is C there. So that wasn't really useful. I'll tell you, let me skim through these really quick. Let me look for an actual genetic. Here we go. Here we go.
I have a question, Professor.
Yeah. Go ahead, April. Apparently my dogs just took umbrage at your request. Buttercup.
Buttercup, aw.
Buttercup. Yeah, she just got her surgery. She just got her tube removed and she's like back to her old self already. It's kind of amazing.
I love that.
It's like the only difference is she wears a shirt now. So she doesn't lick her wound.
Oh, that's good, yeah. Better than the cone.
Yeah, oh God, the cone of shame, we call it.
So for Mendel's, do we have to do Punnett squares for that? Do we not have to like switch
For this, for the one that I have here or on the test?
on the test?
Well, I would say some questions will require you to do it to get the answer, but you won't have to present it as a answer itself.
Okay.
So I would definitely say have your scrap paper handy for this test. All right, so here we go. Imagine a species. You guys want me to do my my British accent when I'm doing this? I read it every night in my British accent, or would that be grating?
Yeah, let's do it.
Imagine a species with 3 loci thought to be on the same chromosome. The recombination rates between locus A and B is 35%, and the recombination rate between locus B and C is 33%. Predict the recombination rates between A and C. I'll do it too. All right, so I see 68%. So here's how I would do this naively. So we got the between locus A and B is 35%. And then they say between B and C is 33%. Okay, so we can see that B and C are closer together than A and B. I don't. Yeah, so we're gonna have to do this. Right, and so the answer would be the addition of those two, which would be what, 68%. So when they say 2%, what do they mean by that? So imagine it was like this. Hold on. So if A and B are here like this, and B and C, let's see what C's recombination is 33%. If C was here, right? Here, let me draw that bigger. So one possibility would be this, where this is what, that was 35 and this was 33, right? So this to here would be 68. But this is also possible. That's the key. So the distance here would be 33. The distance here would be 35. And so this delta would be 2. So it's got to be either 2 or 68. And so, by the way, like what we have to do therefore, what would be the next experiment have to be? What would the next cross be to really hit that home? If I asked you to propose another cross to distinguish between these two possibilities, what would that cross be?
Would it be based on location?
So let me ask it a different way. With A, B, and C as possibilities, which two across that investigated two loci, what would we need to do? What, in other words, what piece of data is missing here? What piece of data don't we have?
The recumbent for A and C.
Exactly. Right, so we would have to do that to prevent the confusion between this possibility. Let me draw that without letters over, right? The choices are either this or this. And the only way that you could distinguish those two is by doing this cross, by crossing it so that we see recombination by basically doing a cross analysis, calculate the recombination frequencies between A and C. All right, good. So what? That's not. I don't think that might be a mistake. They never make mistakes in these slides.
I don't get it.
Yeah, me neither. Me neither.
Me too.
Like there's no math you could do to get 50%.
Oh, hold on a second. I think we're all making the same mistake. We're making a rookie mistake here. Let me think. No, that's not the mistake.
Oh my gosh, it had to either be A or C because we're predicting the rate between A and C.
Let me see if this problem exists somewhere on the internet. I have to unshare the screen to copy the text from the PowerPoint. There we go. Oh, come on. All right. Where? There, this one. Let's see. No, yeah, Morgan's experiment. Nope. There's so many websites that have this cheaty stuff on it. Yeah, there's no, I'm sorry guys, this
Can you share the question again? I might be able to find it.
Yeah,
on the-
yeah, I'm gonna, yeah, I'm gonna share. There you go. I think that's just wrong. You subtract them out. There's no possible way you can do any math here to get a 50. Yeah, you know what, guys? I think this is just,
There's a Quizlet and it says that the answer is C.
yeah, no, and that's because that Quizlet was a student pulling this, or a student taking this and just putting it up there. So anything you find on the internet is going to be derived from this bank. So they're all going to have the same answer. I'll tell you what, let me after class, I'll do some more research on this, because I would, if I was given this, I would say A. I mean, there's no other possibility. This is just wrong. So
Do you think maybe it's something along the lines of like if you add those two together, it's 68%, but like maybe it's not like exactly 68%. So it's like less than 68, but greater than either of those, like the 33 or the 50. But I guess you could say that's 39 too.
yeah, no, I see what you're saying, and it turns out this is all linear. That's what makes this so accurate, because these genes are like, they are literally mapped on the chromosome. So if this is 35, then and you know, there's a whatever, there's another gene over here, 35, and this is 10. When we do the experiment for this, that will be 45. Like, it's always like, the math always works out for these experiments for fruit flies. So I don't have a good answer. So let's just stop wasting our time on this. I'll dig into this and I'll send you a Zoom message here. Which is type of chromosome alteration that differs from all the others? Oh, that's here. There's a conceptual little fun puzzle. Which one of these differs from the others? And I understand, Julia. Okay, I see one answer. Anybody else want to? Everybody's just gonna say whatever Julia said. Or B. Oh, okay, I like why you think B might be too. Okay, so triploidy, 3 sets of chromosomes, tetraploidy, 4 sets, octoploidy, 8 sets. All of those are examples of polyploidy, right? And so aneuploidy is the wrong number. It's just a generic catch-all phrase. All right, good. All right, and Drosophila, that, by the way, that's how you say that word, Drosophila melanogaster. In Drosophila, white eyes are due to an X-linked recessive allele, XW. Which of the following crosses could not result in a white-eyed Drosophila male? That's a great question. Go ahead, see what you got. Feel free to, you know, use your paper if you want. Okay, ready? The secret to answering any question that deals with X-linked traits is the notion, right? Here's the insight, that the males always get the X chromosome from mom, right? So the only way a male could be white-eyed is if they receive that recessive allele from mom. Okay. So mom has to have a recessive allele. So if mom is homozygous for red eyes and dad has white eyes, kid can't have it. Oh, you know what? Let me say that a different way. The male always, oh no, I guess I said right. The male always gets the X chromosome from mom, not necessarily the gene, right? Because if it's a male, it has a Y. And the only way you can get the Y is from dad. So dad's donation is the Y. So all male X chromosomes are maternal, right? My X chromosome as a male, right? The only way I can pass on any genes that I, Andrew Apolito, have in my X chromosome is if I have a female daughter, right? Because my sons all have my Y chromosome. So my X chromosome ended its lineage. Maybe that's why I had that biological imperative of having a daughter, because my X chromosome is like gasping for breath and be like, we're dying here, we're dying, we're going away. And so after I die, that X chromosome's done, right? That's the end of it. That's kind of sad. Man, I need a daughter. Just kidding. Anyway, so what's the answer?
It would be A.
Right? Okay, good. Next.
Wait.
Let's see. Whoa, what? Sorry.
I have a question also, because with that, I was thinking more on the lines of like, if it's hetero, you would have a box that would be like the both recessive genes. So I was thinking that like if it was like a heterozygous, then you would have one box that
Wait.
would like leave the option for them actually getting it.
So
Oh,
look,
we
here
don't
we go.
want to-
So here's the mom, right?
Yeah,
Here
that's what I
would
was
be
saying.
the
That's why I
heterozygous.
didn't pick it.
Oh,
But I
that's
was
why you-
wondering if that was like, if there was a question on the test, if I should look at that differently than that way.
All right. I'll tell you what, Angela, do me a favor. Repeat your logic and let me hear it again.
Okay, so like when you're doing the Punnett squares
Right.
and you do like a heterozygous coupling, so like one capital and one lowercase, one of the squares for like both of the both of the partners involved, then one of the squares is gonna it's usually the lower right one is gonna be like the tiny x's, like the recessive one.
Yeah. Okay. So whenever you have a situation where it's not sex linked, you're thinking about the classic monohybrid cross, right? You're thinking about, this kind of cross,
Yeah.
right? Yeah. The bottom left that'll always be the special one.
Yeah.
Yes, that is a good way to think. Now, so when you're doing a sex-linked cross, when you're tracking the sex chromosomes, there is no magic bottom right. Instead, both of these bottoms are the males and the tops are the females. See that? So like these are the females and these are the males.
Okay.
See that? Right. The XY, right? Female. So you can kind of, there's the traditional monohybrid cross. So think about autosomal traits versus sex-linked traits. So sex-linked traits are going to have a fundamentally different kind of Punnett square.
Okay. I guess I just got lucky for that question then.
Yeah. Okay, next question here. What's the relationship between recombination frequency and the physical distance between genes on chromosomes? The closer two genes are, the lower the recombination frequency. The further apart genes are, the lower the recombination frequency. There is no relationship. All genes have the same fixed barring recombination frequencies. Or D, there is no relationship. All genes have random recombination frequencies. Okay, I see some. Okay, good. You're correct. The closer they are, the lower the chances, right? And so you can take with the little ruler I have here, right? So as I move these away from each other, the chances go up and the chances go down. Good. To the point where if they're so close together, right, we'll see those traits almost all the time together. Good. Like red hair and freckles. Okay, red hair and freckles in humans, those are linked genes. very close together on the chromosome. All right, let's see. Oh, here we go. Here's a math question. What is the expected recombination frequency for a test cross between the cinnabar and vestigial loci? That requires a whole set of skills here. So I'll give you guys a minute on this one. So like, where's cinnabar? There it is, cinnabar. So we have the cinnabar, which is here, and the vestigial, which is here. Right, so what you're doing is you're seeing between here and here, we got 57 to 67, right? Or I'm sorry, 57.5 to 67. So it's almost, but not quite 10, which means this makes the most sense, right? And so the answer is sure enough. Oh, phew. All right. Did the concept here make sense? Right. Okay. All right. Here. What's the expected recombination frequency for a test cross between short aristi and vestigial? So short aristi, that would be the first. Where's my mouse? There. This guy here to vestigial. So that's right. That better be 67 or I'm going to be upset. Oh, wait. I think that we have like a troll at Pearson who's just messing with us at this point. That doesn't make any sense. Let's look carefully again. Okay, short aristae and vestigial. Right, there is literally no way that could be 50%. Unless, all right, I have an itch. I have a mental itch, which means I remember learning something long ago and I can't quite remember, but here's the possible explanation. I'm just going with the itch here, that there reaches a point where you get far enough apart where it's not linear anymore. And so maybe that point of no return is 50%. But no, that doesn't make, like, in other words, you can't get recombination frequencies in a single experiment any greater than 50%. Don't write that down. I'm just spitballing here. I want to go back and look at the chapter and dig into it. But I
have a question.
Hold on, I'm just processing this mentally. The chance can't be greater. If it happens all the time. Okay, go ahead. What's your question?
So if you went from the short aristae, I guess, and then to the brown eyes, it's 104.5%. Is that possible? Like to have more than 100%?
Well, that's the expert. This is a calculated number based on all the various crosses. So no, you can't have a higher than 100% chance of anything happening. That just means always. So really, you would say that these two traits are not linked. Right? That's a key, and your book does mention this. When Mendel was doing his experiments, it turns out two of the traits he worked with, I forgot which two they were actually on the same chromosome in peas, but because they were so far away from each other, recombination always occurred, and so he didn't perceive them as being linked. Right? So linkage is not necessarily always be on the same chromosome. You have to be close enough where you sometimes come along together, right? So that you can think of them as being physically linked on the same chromosome, but behaviorally unlinked based on crossing
So
over.
you're saying that it wouldn't be 104.5%.
Yeah.
So that
If the question said, what's the recombination chance between these two? I would say 100%. It's always going to happen. You can't say something has a greater than 100% chance of happening.
So maybe that has to do with the fact, like the ratio then, to the 57% that we think it is, because we're doing it out of 104.5, not out of 100.
Oh, that's no, we're not. What we're doing is we would in the actual experiment, we would count the number of offspring, and that number could be whatever, like there's 37 flies or there's 4,372 flies. Go ahead,
So
April.
the last one said 50 as well. So maybe there's like a cutoff point at 50%.
That's what I was thinking. That's what I was saying. Like greater than 50% just gives you a 50%. You can't have a greater than 50% chance. But so like I was thinking in my head, if we do that, if we have 2 loci here, right? And so that's either going to happen or it's not going to happen. And the percent chance that happens at any given moment is 50-50, the cutoff. This is where my mathematical skills fail. So let's see if there's something about, there we go. Okay, this is from some like advanced, this is MIT's Department of Biology, introductory biology class. Now I feel embarrassed. All right, here. Oh, here. Why is the highest possible recombination frequency 50%? The recombination frequency between 2 genes is equal to the proportion of offspring in which a recombination event occurred between the 2 genes during meiosis. The recombination frequency between 2 genes cannot be greater than 50% because random assortment of genes generates 50% recombination. Non-linked genes produce, okay. If you think about 2 genes on any, 2 separate chromosomes, you're always going to get 50-50 because of independent assortment. So you can't ever get greater than 50%. You can't get greater than not linked. There you go. And I'm embarrassed to say that I haven't dug this deeply into crossing over since I took genetics. And this is, yeah, here, I'll post this in here if you want to dig around on this website. This looks like a really useful website, actually. I'll put this in our persistent Zoom chat.
I was gonna say, is there a way you can send us like
There it is. Yep.
link? Are there more for like the other chapters, or is it just for this chapter?
I just did a random web search. I typed in recombination frequencies greater than 50%, and I looked at the top hit. So, and this is something. So in the past, when I've taught this, I used to tell my students that don't worry about the details of this stuff. I wanted you to get the concept of crossing over and linkage, but nothing too detailed in here. And then as I've gotten more comfortable teaching the more advanced versions of this, I've leveled up a little bit myself. And so that for me was just me refreshing my memory on something that, remember that I told you I had an itch earlier. So it can't be greater than 50%. And that's why that makes sense. Okay, so that goes back to this question here. That's why it's got to be 2 or 50%. Okay. I feel so much better now. My afternoon class is going to have no knowledge of this ignorance of mine. You guys can chat with them, be like, just to let you know, he didn't know this morning. All right, it's 12:55. Let me oh, by the way, I dumped this PowerPoint into your folder for OneDrive, so you can pull this up if you want. Yeah, you can play with these some more if you want. Okay, karyotype for monosomy. Yeah, so I tell you what, I won't, or I'll tell you what, I'll go through these slowly, and if you want, you can play this back later and pause it at the screen for the question, and then you can see if you got the answer right. Right. So I'll just tap through these quickly. Which of the following diagrams best represents a karyotype of a monosomy? And so this is, so in the video, if you're watching this in the future, pause it now and take a guess, and then I'll give a second and I'll give you the right answer. Okay, so B. Okay, next one. Non-disjunction can happen in either meiosis 1 or meiosis 2. Consider n+1 and n-1 gametes involving chromosome 21 that are formed from non-disjunction of meiosis 1 and meiosis 2. Select the best comparative statement. I'm not going to read all these, so pause it now. And here's the right answer. D. Oh, and that's the last question. So I skipped a bunch. I'll go through very slowly now. And I'll just do this. So again, pause it if you want. to not know the answers. And again, we said earlier, you can't get greater than 50% because unlinked genes give us the ratio of 50-50 because of independent assortment. Same reason. Okay, here, this is the new one that we haven't seen yet. So in tomatoes, the heterozygote plant with normal fruit and purple stems is crossed with a recessive plant having fasciated fruit and green stems. I have no idea what fasciated means. That's a new word for me. The following distribution of offspring is observed. And then normal fruit, right? So we have all these different numbers here. What conclusion could be made regarding the loci for fruit shape and stem color? The loci may be on the same chromosome more than 50 map units apart, or they may be on separate chromosomes. The loci are on the same chromosome, 23 map units apart. The loci are on separate chromosomes. The loci are on the same chromosome at an unknown distance from each other. This is a great question. So the way I would carry this out, so I'll pause, and if you want to try to work this out on your own, go ahead, pause the video now. Okay, so my explanation would be, so here's the key. Do you see how the numbers lump into two types? And so the lowest ones, those are always going to be the recombinants. Always, right? Because if it's 50-50, then they're all unlinked. So the idea here is you would have to add these together, right? So what does that give us? That's 11 plus 11, 22, that's 23%, right? That's the secret to these. That's one of the little puzzles that people mess up. They'll say 11.5 map units. That's 23 map units because you have to add up all the recombinants together. Okay, so this tells us that these two genes are 23 map units apart. It's under 50%, right? So the answer should be B. Thank God.
Can you
All right.
Google that really quickly?
Next question. All right, so same setup. I'm not going to repeat saying it. Just so notice that it's different plant, different pair of traits, but we know where we're getting at here, right? And you see how we see 25% across the board. That tells us either, right? So this isn't necessarily, you can't say, where's my mouse? It's a little dot, it's so hard to see here. This can't be the right answer, even though you want it to be. They're on separate chromosomes, obviously, because it's independent assortment. But the thing is it's also possible, oops, not that one. It's also possible that they're on the same chromosome, but they're greater than 50%, 50 map units apart from each other, right? Because the behavior of two genes on the same chromosome greater than 50 map units apart, behaviorally, these genes cannot be distinguished by two genes that are on completely different chromosomes because you still get a 50-50, right? So that's why the answer here has to be B. Okay. So again, either they're on different chromosomes, and so you're going to see even distributions because of independent assortment, or they're on the same chromosome, but they're so far apart from each other that they behave as if they're not linked. Okay. All right. And then, we got to these already. Which of the following diagrams depicts a karyotype of monosome? And we're back where we started on this. So I'll end this PowerPoint here. Or here, I'll give you the answer to that. Just that last one is B, right? And the reason for this is, right, all the others here. So here, if you look at the first scenario, you see there's 3 copies of one of the large chromosomes and 3 copies of a small chromosome. So this cell here, this would be a triploid cell because it's got an N of 3, and this is I'm sorry. It's got an N of 2. How do I know that? so you look here, there's two visually distinguishable types of chromosomes. There's a big one and a little one, right? This chromosome here is different than this chromosome here. So there's two, so that's an N of 2. And there's 3 copies of each. This is 3N equals 6. So that's a triploid cell. Okay, this cell here, again, it has an N of 2 and it has 3 chromosomes. So this is a 2N minus 1. You see that? Because there should be one here that's missing. This cell, this is a haploid cell with an N of 3. Easy peasy. Those are 3 different visually distinguishable chromosomes. And then this here, this is an N of 3, and this is a 2N equals 6. Right? So this is a traditional diploid cell. So if this was a normal body cell, we would say this is the euploid number, right? This is euploid. This would be euploid. Because those all look normal. This is the one that's aneuploid because there's something wrong with it. Okay. All right, cool. And then let's see. This is just so you can, I'm not going to walk through this one. There's just too much on the screen right now. I'm tired and we're done. And that's the end of it. Okay. So we'll say why is it not letting me? It's interesting. Anyway, I wasn't able to draw on it anymore. Screen share close. And I'm gonna stop the recording now. See you next time, recording people.